to check out if you're Did you get it? No? No, I can't. Let me see. Good morning to all of you. It's a pleasure to be here today for the Mini-Symposium, Aging and Future Challenges. This is a joint of two groups working together. So the CAPS Print funding this event, and we have also the Institute for Advanced and Congregated Studies on Aging, that's represented here from Professor Maísa and myself. So IEAC is this institute from Unifesp, and CAPS Print is providing the financial support for this event. And I'm very happy because today I have here 4 good representatives of the aging studies. So we have Professor Daniela Frasca from the University of Miami. She has been here already in 2019 before the pandemic situation, and she's working on B cells, aging, obesity, and several inflammatory conditions. I'm very happy also to have here Professor Maísa Senderolo. She's working with geriatricians, so all patients healthy with 80 and more years old. So she's congregating all these health conditions, and how can you get there and keep yourself there is not easy, is it? So she's gonna talk for us about that. And we have Professor Miriam Jasulionis. She's working on epigenetics, not really on aging, but on cancer cells. So she's gonna give us a good idea how the cell can go, how the whole organism can go on aging process. And we have also Professor Thiago Rodrigues from Federal University of ABC. He's gonna talk to us about mitochondria, which is a very important field, just now starting to getting people working on it. So once more, thank you all for being here. And I think we're going to start with Professor Daniela Frasca. And thank you again, Daniela, for being here. Thank you, CAPS Print, and thank you to the ESC Institute and Unifesp for supporting us on this event. I hope you can enjoy it, and it's open for questions at any time. You can stop and ask Daniela something, or you can leave for the end of her talk and ask everything that you like. Okay, thank you very much.
Thank you, Valkyria, for this very kind invitation. I'm happy to be here again after the pandemic. I'm very happy to be again in this room and to meet a lot of you, either in person or through internet. I will talk today, first of all, of one of my major interests in research, which is obesity, and how obesity is inducing an acceleration of the aging process, at least for humoral immunity, which is antibody responses to infections and vaccines. Now, let me get familiar with this. Probably I have to close this page and open the other one, right? This is the pointer, right? Okay, so the title of the first presentation of today is Obesity Induces B-cell Immunosenescence, so accelerates the aging process for humoral immunity. Doesn't like to work. I'll do it from here. Thank you, Valkyria. I need to get familiar. So this is my thank you slide for the people in the lab that have performed all the experiments that I'm going to present today. And a special thank, of course, to the doctors, without the help of whom would have never been possible to put together the results that I'm presenting to you today, especially Dr. Thaler, who is the director of the Division of Reconstructive Surgery, because he's very keen in research and is very helpful in giving us the possibility to use large number of samples from people undergoing weight reduction surgeries. And of course, facilities at the University of Miami and funding, very important. Okay, so this is what I'm going to talk to you today, which is my main research focus nowadays, and is this topic: is obesity really a mechanism of immunosenescence for humoral immunity? I'm going to show you how obesity is inducing defects in responses to infections and vaccines, and at the same time is inducing autoimmunity, so good and bad antibody responses. How is the adipose tissue contributing to these effects of obesity on humoral immunity? And this occurs through a redistribution and readjustment of the peripheral B-cell pool in individuals with obesity. And finally, I'm going to show you some recent data showing how obesity is not only responsible for B cells that are inflammatory, but these B cells may actually polarize other immune cell types towards a pro-inflammatory phenotype. Okay, I don't have to tell you how obesity is becoming an epidemic worldwide. These are data from the CDC, so these are data from the United States, and these are trends in age-adjusted obesity preference, and not only obesity, which is BMI over 30, but also severe obesity, BMI over 40. This is something very bad, but something very helpful for me to do this research, because Miami is an area where we have a lot of obesity, prevalence of obesity. So it's kind of easy to recruit patients actually, individuals for our studies. Everybody knows that obesity is associated with a lot of health problems, not only insulin resistance and inducing type 2 diabetes, abnormal cholesterol levels, but has a lot of problems at the level of the cardiovascular system, respiratory, gynecological, gastrointestinal, increased infertility in males, musculoskeletal problems, and also is associated with the incidence of cancer and also incidence of metastatic cancer. So how is obesity impacting the immune system? So which are the immune complications of obesity? Well, the immune complications of obesity start with these things. So obesity is an inflammatory condition that is regulated by induced and then maintained and regulated by the chronic activation of the immune system. Both the innate immune system and the adaptive immune system becomes activated chronically. And this leads to local and systemic inflammation, which contribute to a lot of pathologies, which is typical of old age. Here I'm just saying type 2 diabetes and autoimmunity, but also cancer and many other conditions. Obesity is associated with increased probability to get infections from bacteria, viruses, and fungi. And this has been shown with Staphylococcus, the Streptococcus pneumoniae, Mycobacterium, H. pylori, the recent coronavirus responsible for the pandemic, influenza and Candida. Obesity is associated also with reduced responses to vaccination, like influenza, hepatitis B, and COVID vaccines. In general, there is a decreased response of immune cells upon vaccination, upon stimulation in vitro or in vivo. Obesity accelerates thymic involution, and for what I'm very interested in, there is a decreased capacity of the immune response to make protective antibodies against, neutralizing antibodies against infections, and also antibodies against vaccines. And there is an increase of pathogenic phenomena like the secretion of autoimmune antibodies. So all these characteristics are typical of aging, of the aging immune system. And for this reason, all these areas of overlap brought us to the consideration that maybe obesity can be a biomarker of accelerated aging. Again, at least for humoral immune responses, but not only. It seems very clear that it is also for other immune responses. So when we studied during the recent pandemic, the response of individuals that are lean or obese to the infection with the SARS-CoV-2, and we measured the neutralizing antibodies in serum, we found that only lean individuals were able to make neutralizing antibodies. But these people were not only less in protective responses, but they were inducing a lot of autoimmune responses. And here I'm showing two specificities, MDA and AD, that are associated with increased fat mass. And I will show you later which are the reasons why I'm measuring this. This is a product of lipid peroxidation. And this is a product of cell death in the obese hypertrophic adipose tissue that occurs in individuals with obesity. So you see this significant increase in autoimmune antibodies, which are also evident in individuals recruited previously in the pandemic, because obesity is not an autoimmune condition, but is associated with a lot of autoimmune pathogenic antibody secretion. Of course, there are many other specificities that are released in the serum in individuals with COVID. And for example, we have antibodies specific for type 1 interferon. So these people affected with this virus were able to shut down with these autoimmune antibodies the natural responses against the virus, either systemically or locally in the lungs. And this is for infection. But when we measure the response to the influenza vaccine, and we measure the protective titers, which means this is the reciprocal of the titer measured by hemagglutination inhibition assay, which is the best correlate of vaccine protection. We found that the titers were decreased in obese individuals, both young and elderly. But the most interesting finding of these results was that the response of these two groups, the young obese and the elderly lean, were absolutely overlapping. And this, for the first time, made us considering obesity as an accelerator of the aging process for antibody responses. Now, the same serum samples of these individuals were tested again for two autoimmune specificities associated with obesity. And we found that obesity was able to increase in both young and elderly individuals the secretion of these antibodies. But again, like here, these two groups were giving similar responses. So the young obese and the elderly lean look absolutely overlapping. So it is probably true that obesity accelerates age defects in humoral immunity. At that point, we didn't know which the mechanisms were. So we started evaluating the human adipose tissue. And as I told you, we got a lot of samples from individuals undergoing weight reduction surgeries, mainly breast and abdominal anecholectomy surgeries, which is subcutaneous, which is very, very inflammatory. I'm not talking today of the visceral adipose tissue that it comes from heart surgeries. And from these patients, we get 3 different samples from the heart. So today is I'm only focusing on the subcutaneous adipose tissue and mainly on the breast, which is less inflammatory than the panus, than the abdominal adipose tissue, but still very, very inflammatory. So what happens with age with the adipose tissue? Well, there is an increase in the fat mass. From the prenatal period, where the adipocytes, which are the non-immune cells of the adipose tissue, get formed, there is an increase of the number from birth up to 10, 15 years of age, approximately in humans. But after that age, the adipocytes don't increase in number, only in size. They become hypertrophic. And more they increase their size and more inflammatory they are. And this is something that happens also in individuals that are thin, that are not obese in later ages. So this is the first thing, an increase with age in the fat mass. But there is also a redistribution of the adipose tissue, depots in aging, with an increase in the visceral, with an increase in the visceral mass as compared to subcutaneous. These are several studies showing this, and this is the ratio visceral divided by visceral to subcutaneous in males and in females. You see there is this significant increase in age in visceral versus subcutaneous adipose tissue. This is white adipose tissue. And a very interesting thing is that, and this is something that, this is evaluated only by BMI, so the body mass index. But actually, when you go inside the body and we take some body measures, we can see that at different ages, this is a young person, this is a person over the age of 65, so a typical elderly individual. You see how much fat there is inside. This can only be diagnosed by CT scan and MRI. So it's not something that is usually done by physicians that have to refer a patient for some kind of analysis. And this phenomenon is very frequent and is called now TOFI. TOFI means thin outside, so low BMI, fat inside. And this reflects the deposition of fat, triglycerides and other kind of fats in internal organs, starting from the blood vessel, but ending on the heart. We have a lot of fat on the heart, on the epicardial adipose tissue, but the pancreas, the liver, the skeletal muscle. So the body gets a lot of fat with age, which is distributed internally. And sometimes we don't know if a person that is aging apparently well is really well because doesn't have enough fat inside, or it has and is not visible. So now, at least in the United States, the doctors, geriatricians are referring the patients to do some CT scan, not MRI, which is more accurate, but the CT scan are not very expensive. And even a technician, not a doctor, can evaluate this difference in the amount of fat. So the result is plus, 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 minus. Is not exactly telling you how much fat is present on the visceral depots, but at least it's some information that we can get from these patients. So what is the adipose tissue doing? The adipose tissue is an immune tissue. Now it's very clear. Before it was only considered a storage of nutrients. Well, actually not. It's an active immunological tissue. And actually is, for what we have shown, is an active chronic site where autoimmune antibodies are secreted. What happens in the adipose tissue under obesity conditions? We have hypertrophic adipocytes. These hypertrophic adipocytes are highly secretory. They secrete a lot of inflammatory compounds. First of all, TNF-alpha. TNF-alpha induces lipolysis, is the best inducer of lipolysis, a chronic activator of lipolysis, which is the hydrolysis of triglycerides that generates free fatty acids and glycerol a lot in the tissue. Of course, free fatty acids and glycerol go also in the circulation and can affect distant insulin-sensitive tissues like the pancreas, the skeletal muscle, etc. But locally in the tissue, they activate macrophages, resident macrophages, together as other immune cell types that have the receptors for fatty acids. And this leads to the inflammasome activation and NF-kappa B activation, ROS secretion, and an NF-kappa B activation is leading to additional secretion of inflammatory cytokines. So this is a vicious cycle of inflammation inside the tissue that allows secretion of several inflammatory compounds, then released in the circulation that can affect other tissues and organs of the organism. I told you this, the free fatty acids can go, for example, to the beta islets in the pancreas and stimulate additional secretion of, for example, chemokines that are released by the adipocytes in large amounts, as well as by the immune cells, but also by cells that are in other tissues and organs. And of course, these chemokines released bring to the recruitment of a lot of immune cells from the blood into the adipose tissue. Now, the blood is the blood of a person with obesity or super obesity, so is already enriched in inflammatory immune cells. All these infiltrate massively the adipose tissue, and you see we have every possible kind of immune cell that contributes with the secretion of other inflammatory cytokines and chemokines, as well as many other things. And because we have an accumulation of inflammatory B cells in addition to other inflammatory cells, look at this, the CD4, Th1, and Th17, and we have a lot of T gamma delta, because this is a tissue full of lipids, so the T gamma delta should be there to help B cells to make antibodies against the lipids. So I'm going to show you now, just in the next cartoon, that there are every kind of cell type is actually present in the adipose tissue, infiltrating the tissue from the peripheral blood. So we have B cells that are increased in the adipose tissue. We have T cells and their subsets, CD4, CD8, gamma delta 1, gamma delta 2, every kind of T cell. NK increase, whereas NKT are reduced in the infiltration. And of course, a lot of monocyte and their subsets. This is the major population of immune cells in the adipose tissue, made by the resident, the ones that are there since the time that we were born, and those that have differentiated in the tissue from circulating monocytes. So in many years, we have characterized the pathways responsible for the secretion of autoimmune antibodies in the adipose tissue. So first of all, what we need is the availability of this autoantigen in the tissue in order to make autoimmune antibodies. And these autoantigens are made available by several pathways and processes that are chronically activated in the tissue. One of these is lipolysis. I just told you that lipolysis releases free fatty acids, which are immunogenic. Palmitate, for example, is an autoimmune specificity that B cells make antibodies against. But there is hypoxia. Why hypoxia? Because under the obesity conditions, the adipose tissue increases enormously in size, but there is not a concomitant increase in the vasculature. So there are a lot of areas of hypoxia, microhypoxia, which are responsible for cell death. Cell death means the adipocytes, together with the immune cells that are around the adipocytes in these so-called crown-like structures. So everything dies. And these cells release what they have intracellular, whatever is cell-associated. Some of these things are known as autoimmune antigens. For example, DNA, RNA, single-strand, double-strand DNA is released abundantly. But there are also other proteins that I will show you now that are not known as common antigens. But you can find specificity against these proteins can be found in the serum of individuals with obesity. Once the antigens are released, protein and non-protein antigens, lipoproteins as well, a lot of lipids and droplets are released, they are presented to the proper way to the immune cells in order to make antibodies. So we have canonical proteins presented to the MHC class II, and other proteins that are presented by the adipocytes. Yes, the adipocytes, through the CD1b, can present antigen. And of course, the T cells that contribute to this antigen presentation are different. These are typical CD4, these are gamma delta. So that's why there is this humongous influx of gamma delta that can either be recruited from the circulation or can also differentiate locally because they participate in the antigen presentation. So this is a secondary, the adipose tissue is a secondary lymphoid tissue. In some cases, a tertiary lymphoid tissue. So there are germinal centers, B and T cells. These collaborate very well, and they allow B cell maturation, differentiation, and process from B cell memory into plasma cells able to secrete. So what do they secrete? Which is the antibody specificity that is secreted? Well, is what we call adipocyte-specific IgG. Because in these conditions of hypoxia and cell death, the cells, adipocytes and immune cells, release whatever is their content. And these proteins altogether stimulate an immune reaction. So we have characterized, and I'll show you in the next slide, more than 4,000 protein specificities. Actually, there are more and more and more. There's the group of Daniel and Sean Weiner, now in Stanford, that again, actually one is in Stanford and one is in Los Alamos, that have characterized more than 9,000 different specificities that you can use in a protein array, extremely expensive, and check the serum samples from individuals that are either insulin resistant or insulin sensitive, young and old, people with diseases versus people healthy. So it's very important to know the specificities. Of course, we don't know each individual which specificity is making. But the interesting thing is that, you see, we get a lot of tissue removed. So this is a good reason to be in Miami. We do collagenase digestion of the tissue. And after the collagenase digestion, we get the majority of the immune cells in what we call stromal vascular fraction, which is down here after centrifugation. And the adipocytes, the floating adipocytes, are here. So we can have the separation of the two fractions pretty well. You see, you don't need to stimulate these cells at all, because the chronic activation of all the processes that I showed you before is responsible for the secretion of these antibodies. If you stimulate with any toll-like receptor the T cells, and these antibodies that we spontaneously secreted can be found, these ones secreted in vitro, can be found in plasma. So actually, from the adipose tissue, we generate a response that can be detected in plasma, not only our obese individuals. So elderly people have the same specificity, actually. So when we try to characterize the specificity with the HPLC and mass spectrometry, only of the specificities for IgG antibodies, because IgM is a totally different story, very complicated. And we found at the end, out of more than 2,000 proteins and peptides, 16 specificities that were associated with the majority of our donors. More than 80% of donors were showing these specificities. All of them cell associated, either in the plasma membrane, in the cytoplasm, or in the nucleus. But they were, some of them, for example, histones, were known already as autoimmune specificities. But many of them were not. So there are signaling proteins, there are proteins involved in cell metabolism that become immunogenic just because of the structure and the condition of the adipose tissue. Is not something, well, of course, if you take the FIN, tyrosine kinase, and you put it in vitro in the presence of immune cells, there can be some response in terms of antibodies, but this occurs mainly in the adipose tissue. So it's the condition of the tissue that allows the secretion of these antibodies. And this is what we are doing right now, using this panel of 16 proteins to test the individuals that have comorbidities in addition to obesity and type 2 diabetes. Okay, so this is one thing. So the adipose tissue leads to the secretion of these antibodies. But which are the B cells that make these antibodies in the adipose tissue? So I, in particular, had a very particular interest in this subpopulation of B cells, because these are the most pro-inflammatory B cells. They are the ones that develop spontaneously in conditions associated with inflammation, like healthy aging, obesity and type 2D, every kind of autoimmune disease, chronic viral HIV, parasitic infections, malaria, and also recently in COVID. So these are the cells that were found in the lungs of people affected by acute COVID as well as in the circulation of these patients. So this is the way that we usually evaluate B cells in the circulation or in the tissue with these two markers, IgD and CD21. And these cells are called double negative just because they don't express any of these markers. And this is now an international recognized nomenclature. Before they were called in several ways. Okay, so B cell differentiation starts from naive B cells, and then there is the generation of memory. IgM first, switch memory, which are the secondary antibodies making cells, and then double negative, that for a long time were considered just to be cells exhausted or cells prone to cell death. Not true. They are extremely active transcriptionally and metabolically. So in order to be active transcriptionally, in an environment which is really hostile, like the inflammatory environment of the adipose tissue, they have to do a metabolic reprogramming in order to survive and do their job, which is secretion of inflammatory markers and autoimmune antibodies. Okay, this double negative, of course, don't make protective antibodies, but they make autoimmune antibodies. And they make autoimmune antibodies in any condition associated with autoimmunity, rheumatoid arthritis, lupus, Sjogren, pemphigus, and also in chronic infectious diseases. They make pathogenic antibodies in HIV and malaria patients and in COVID. They are very inflammatory, and there are markers associated with immune activation and immune senescence to identify them. And as I have anticipated to you, they have to become hypermetabolic to survive and do their job. Okay, these are the double negative cells you see by flow cytometry using the 27 IgD markers gated on live CD19 cells. Significant increase of double negative in elderly, healthy elderly individuals. Very similar to what happens with obese. This is the staining of the blood, peripheral blood, from a lean and an obese individual. You see the increase in frequencies and numbers also of these cells, which again is absolutely overlapping to what we see in elderly lean individuals. Lean based on BMI, yeah? So that's another thing that I haven't evaluated at this point yet. So again, this brings us the idea that obesity accelerates age defects in the B cell pool. And when we look at the adipose tissue, at the stromal vascular fraction of these people, this is the blood, and this is the stromal vascular fraction of the same individual, you see how many more double negative are present in the stromal vascular fraction of the adipose tissue. And actually, in the adipose tissue, the majority of B cells have a memory phenotype. Memory means the sum of these three different subsets. And of the memory, the majority are double negative. Whereas in the blood, we have approximately 50-50. The healthy person, the majority are naive. And when we do a cluster analysis, it's very easy to see the decrease in naive, the increase in this double negative together with a switch memory. Not so many changes in the IgM, as you can easily see from here. So this is the redistribution of the B cell subset due to obesity and especially in the adipose tissue. So the double negative cells that come from the adipose tissue, once we sort them, of course, there are not many individuals. It's kind of difficult to sort these cells, but they are the ones that if you leave them in culture for 10 days, they secrete autoimmune antibodies. And these are the only ones that do so. And they do so because transcriptionally, they express the protein, the transcription factor, which is needed for autoimmunity, which is Tibet. This is a prime flow. So first of all, we stain the membrane for flow cytometry with fluorochrome conjugated antibodies. Then we fix and we go inside the cell to do an in situ hybridization with a probe that is specific for the transcription factor. So there is, of course, more RNA for this transcription factor, and of course, more protein if we do intracellular staining with the Tibet protein. So they are increased, double negatives are increasing the adipose tissue compared to the blood, and they make autoimmune antibodies, and they do so because transcriptionally they carry the right transcription factor to do this job. They have the phenotype that is typical of autoimmune B cells. When you have a patient with rheumatoid arthritis or with lupus, the B cell, peripheral B cells are low in CD21 and high in 95 and CD11C. Very interesting thing is that they express other markers associated with senescence and immune activation. And look at PD-1. So when you consider an immunotherapy with an anti-PD-1 antibody, well, actually you're going to hit also these cells, which are very inflammatory. In cancer, these cells also are expanded. So it's a good thing that you can target these cells. And of course, they are more inflammatory. Many markers of immune activation and senescence they express, and more inflammatory they are. And you see that in the adipose tissue, there are several, as compared to the blood of the same individual, that express more than 3 markers. We have evaluated many of them. So the next question that we ask is if these cells recirculate, because it's important. So is the response that we see in the peripheral blood only coming from the adipose tissue or not? And there was this study done by the group of Mike Anker, is a friend in Pennsylvania, in Philadelphia, that showed this is what he uses to identify double negative. So Tibet high B cells, actually, they are not found in the thoracic duct as compared to the peripheral blood. It's only here, peripheral blood, but not in the thoracic duct. And the reason is because everything that circulates in the body goes into the thoracic duct. It's collecting lymph from a lot of lymphoid and non-lymphoid tissues. So whatever circulates, you can find it there. Well, it didn't find these double negative cells. And in fact, they don't express, or they express in a non-significant way, the markers for recirculation, which is the cytokine receptor. So we tested also in our samples, comparing the blood and the adipose tissue. Well, CXCR3 is all over the place. So people, in some cases, some double negative recirculate, and in some others don't. And we don't know the reason why is this. But our double negative in the adipose tissue express the CXCR5, which is the homing receptor responsible for them to be in the adipose tissue. So the majority of these cells do not recirculate. Now, this is what we think is happening. The adipose tissue as a model, just summarizing a lot of results. The adipose tissue is generating a lot of self-specificities. These are also to be included in self-specificity, and as well as co-stimulatory molecules, and a lot of inflammatory cytokines. That all these can activate B cells, in particular double negative, that have the right B cell receptor. This B cell receptor, through signaling of STAT, JAK and STAT, is able to activate the transcription factor responsible for the secretion of autoimmune antibodies. Now, the signals that are amplifying this direct signaling pathway are interferon gamma receptor and IL-21 receptor. There is a lot of IL-21 in the adipose tissue. But also this toll-like receptor 2 and 1 able to interact with free fatty acids, especially some free fatty acids that are working not only as specificities to the BCR, but also as co-stimulatory molecule for the activation of Tibet and secretion of autoimmune antibodies. And I want to show you now, just because this is a recent observation, I want to show you now, just to conclude, some data on that. So I told you already that the obese adipose tissue is responsible for lipolysis and secretion of fatty acids. And this occurs because in the presence of TNF-alpha itself, or the downstream molecule activated by the TNF receptor 1 and 2, may break the structure of the adipocytes. The adipocytes are just a storage of lipids surrounded by phospholipids, different kind of phospholipids, and all these structures is maintained by the perilipin. Once you have activation of TNF-alpha, this structure is broken and these lipids are released. Okay? So what have B cells to do with lipids? Well, B cells can accumulate lipids, and they do so in aging and as well as in obesity. And again, look at the elderly lean B cells from elderly lean individuals and young obese. Again, these two are very overlapping. So this is measured by flow cytometry using this lipid tox. Okay, they accumulate lipids. What do they do with these lipids? They accumulate and they use them in a lipid environment like obesity. This is results published many years ago, confirmed later by many other groups. This is the correlation between the fat mass in kilograms and the amount of fatty acid in the circulation. Any kind of fatty acid is present in the circulation, but the palmitic one seems to be the leader. And in fact, it represents 60% of the fatty acid in the body, and more than 30% is very variable in different individuals. The levels in the blood are regulated because the palmitic acid comes from either the diet, from nutrients, as well as from the lipolytic processes that occur in aging and as well as in obesity and in other inflammatory conditions. So this is not only a specificity for autoimmune IgG, but is also stimulating itself. As I showed you before, the toll-like receptors bind the fatty acid, and this interaction is amplifying the Tibet and the secretion of antibodies. Here is the experiment. B cells from young, lean individuals activated to make antibodies. In the presence of palmitate, they make more, and again, very similar to what happens to B cells from obese individuals. And the same happens in elderly. And this is for several specificities. If we take the double-strand DNA, it's exactly the same thing. It doesn't matter the specificity that we are measuring. And again, they do so because they activate Tibet. So palmitate is able to shift the transcriptional profile of the B cells from protective versus pathogenic response. And he is the prototype transcription factor for immune responses. And I think that with this I'm done. Yes. Oh, I still have another, I have other 5 minutes, okay? So this is the last part of my talk, how B cells are inflammatory, and I showed you this. But can they regulate inflammation in other immune cell types? Because to make antibodies, they have to interact with antigen-presenting cells and with T cells. How do they behave with T cells? So it is well known that B cells, this is from the Weiner brothers many years ago. They show that in mouse models of diet-induced obesity, human obesity, type 2 diabetes, and autoimmunity, the B cells can secrete antibodies that can activate through the Fc receptor macrophages to secrete cytokines, inflammatory cytokines involved in insulin resistance, for example, TNF-alpha. They can also interact with CD4 and induce the secretion of, for example, interferon gamma, which is the most important cytokine for insulin resistance. So we wanted to test the effect of B cells on this pathway. So we took PBMCs, peripheral blood mononuclear cells, and from lean and obese individuals, and we stimulate them all together or after the removal of B cells with a stimulus that is inducing inflammatory T cell cytokines. So only T cell cytokines like IL-17 and interferon gamma. You see that if we remove the B cells from the blood of a lean person, nothing happens. If we remove B cells from the blood of an obese person, the secretion of these two inflammatory cytokines is significantly decreased. So B cells contribute to the secretion of inflammatory cytokines by T cells. And today in the afternoon, I will show you that this occurs to metabolic factors. We also confirmed these results, trying to block in vitro this effect of B cells. And this is what we did. This is the simulation of the PBMCs from obese individuals. If we remove the B cells, as I showed you before, there is a significant decrease in IL-17 as well as interferon gamma secretion. If we put back the B cells, of course, we repristinate, by and large, this response. But if B cells are pre-treated before adding them back with metabolic molecule like metformin or rapamycin, or anti-inflammatory like anti-TNF neutralizing antibodies or vitamin D, this effect actually is abrogated. So you can actually revert this capacity of the B cells in inducing inflammatory cytokines by release of inflammatory cytokines by T cells, just treating them in vitro with some anti-inflammatory compound. Now, we don't have the possibility to do this kind of experiments in the people that participate in the TAME study, which is the study of Nir Barzilai that is treating metformin to treat aging, because we have access only to some of these individuals, and a little amount of blood doesn't allow us to do these experiments. But in mice, we have injected mice with metformin, with rapamycin, with anti-TNF, and we have the reduced capacity of B cells to induce inflammatory B cells and monocytes, monoclonal. And finally, the cells that are treated, the B cells that are treated with these anti-inflammatory, metabolic and anti-inflammatory compounds, of course, they are less inflammatory. So themselves, they secrete less IL-6, P16. These are all markers of the senescence-associated secretory. Okay, this is my summary slide. I hope I have convinced that obesity, by decreasing protective antibody responses and increases autoimmune like aging does, may be considered a mechanism of B-cell immunosenescence. And also, I showed you the data by which the obese adipose tissue contributes to these effects, and how obesity is able to induce B cells that polarize other cell types. I just showed you the T cells, but the same can be shown for monocyte macrophages. Of course, these are characteristic of aging, and obesity is absolutely overlapping with aging, at least for these measures, outcomes. At the level of the individual, obesity is a risk factor for inflammatory conditions and diseases typical of all ages, as I already anticipated: type 2D, cancer, psoriasis, atherosclerosis, and acne. There are several other characteristics of these two conditions together. So the pattern of organ specific deterioration induced by obesity and aging is very similar. Epigenetic modifications induced by aging obesity are similar, and some of those induced by aging can be actually reverted by some dietary interventions. And also the changes in the gut microbiota have very similar patterns. They are not completely overlapping, but they are showing similar patterns. And with this, I conclude. I thank you for your attention, and I open to any questions.
Thank you, Daniela. I'll have the microphone here, so if any of you would like to ask Daniela something. No. So I will ask you something just to start the line. I hope you get some questions after me. Yeah, Daniela, just a curiosity, if you do bariatric surgery or any removal or diet or anything, talking about the blood, can you revert this phenotype, or how much you can revert this?
Yeah, very good question, and that's a dream. because actually we have done the analysis of the blood of these patients before surgery, 6 months after surgery, and not bariatric. I don't have any possibility to recruit patients after they do bariatric surgery. But 6 months and 12 months after the removal of the fat, there is one factor that is very going up and down, and which is IL-6 in serum. And with this up and down of IL-6, these inflammatory markers stay pretty up. So some individuals are going to reduce the amount of pathogenic antibodies that they secrete. They can improve the response to vaccination. They're never coming back to the level of people without obesity. And the reason is that obesity, from studies in mice and also in vitro in humans, is giving an imprinting to the local tissue that is lasting forever. So once you get to this point, these are people with a BMI over 40. So there is never the possibility for them to revert to a condition, even if they lose weight, even if they lose, let's say, 10% of their weight in 6 months and they keep it constant. Actually, their immune response remain with the imprinting of that obesity as stated from the beginning. So some do better than others, but they're never reverting to a condition that is comparable to that we see in lean people.
Daniela, thank you very much for your speech. And okay. I would like to know your opinion about the metformin and the importance as a geroprotective drug, because we have these impressive results for obesity people. So what you can tell us about this?
Yes, I can tell you something from in vitro studies, because actually the study of NIR is not measuring immune cells, immune function. Is only measuring some functional improvement, cognitive improvement, and the concentration of circulating pro-inflammatory cytokines. That's it. So from the cohort in Miami, that is a cohort of individuals, young and old, that have any kind of comorbidity. There is not the possibility to recruit healthy people. So these people with different morbidities and comorbidities, when their cells are stimulated in vitro with the presence of metformin, of course, all the inflammatory responses are decreased. And actually, they do much better when they get the influenza vaccine in vivo. So their HAI response is better. Is not, I mean, in individuals that are treated very early, after the discovery of the diagnosis of type 2D, actually this is what makes the difference compared to those that are taking metformin probably years after they have been diagnosed. But there is an improvement. We published this in 2021. There is an improvement in the a decrease in the inflammatory profile of the B cells. There is a decrease in the hypermetabolic profile of the B cells, because this senescence phenotype is associated with hypermetabolism. So all the intracellular pathways that lead to anaerobic glycolysis, oxidative phosphorylation, have to be high in order to support the inflammatory profile of these cells. And this is reduced by metformin. We have actually very few people as controls not taking metformin. So the comparisons of the study that we published is young and elderly, healthy, type 2D without metformin, and type 2D with metformin. So they come to a point that is intermediate between the young and the elderly without not taking metformin. The ones with type 2D have much lower B-cell responses if they're not in metformin. So it's not too much, but you know, it is in, I think, in Frontiers 2021 published.
Thank you.
Thank you for your presentation. I'm here because I hope to be healthy aging person. And I was quite chattered with the idea that all the markers of healthy aging, thin age, correlates with young obese. So is there any chance of healthy aging?
Well, that's a very good point. Yes, there is. Of course, there are the centenarians, as you know. These are the people that go over the point in which the elderly lean are similar to the young obese. And so by passing that point, they proceed to a very healthy longevity, which doesn't mean that they don't have inflammatory markers, but they have a better outcome of the organism. For example, they have a better microbiota, which is different from the one of healthy elderly, is more between the healthy elderly and the young. And this is one of the best things that I can tell you. And they have also a better immune response when tested in vitro. So the centenarians actually are the real healthy individuals that we have to, use as a good quality of life, let's say, in later ages. So the fact that the B cells are behaving in healthy elderly like in young obese doesn't mean that that person cannot live 90 years in good health and not getting infectious diseases. Is always a balance at the level of the organism. So how much inflammation you have systemically, how much fat you have inside, how good your microbiome is, how good your health and your history, immunobiography, because probably you have never been infected by bad diseases and you have a background of antibodies that can always protect. So it's something very individual. So don't think that the young obese and the healthy elderly, just because they are over the level of young, cannot live a very healthy life. So health span is the goal that we have, not longevity per se, it's just a good quality of life in later ages.
There is help then.
Excuse me?
There is help then. There's hope.
Of course, there is hope. And, I mean, there are so many people over 80 years of age. You do a metabolomic analysis in serum, and they look like people that have a biological age which is higher than what they really have. So it's important how your biological age is more than the chronological. And the biological age is determined by a lot of factors: inflammation, metabolomic, in serum, microbiota, history. The immunobiography of each individual is very important. So which are the stimuli that you have been exposed throughout life that generate your capacity to respond? And also the capacity to get rid of all the stressors that have led to the accumulation of these molecules that can activate the chronically the immune system.
Professor Daniela, thank you for your presentation. My name is Alexandre. I'm a rheumatologist, so I know very few about immunology, but I deal with autoimmune diseases like systemic lupus erythematosus. And I was delighted to hear that there is an array of autoantibodies that would come from cryptic antigens after the lipolysis that they would be available for the immune system. So it's something like lupus. So you have several antibodies, but I wonder, are they really pathogenic or are they only bystanders? This is the first question. I have two other questions. I don't know if you would like to answer.
Let me answer this question.
Okay.
Because I may forget. I'm old as well. So yes, there is the possibility to have specific antigens released in some diseases. When we did our very small protein array to check for rheumatoid arthritis patients, we found half of those specificities in the synovial fluid of people that were diagnosed and were only taking methotrexate as compared to those that they were taking methotrexate plus the biological therapy. And we found a big difference because actually in the synovial tissue, there is a lot of cell death. And the processes that I have shown for the adipose tissue are, I think, we haven't done a deep study, but they may be very similar to those that happen in that inflammatory content. For lupus, I don't have any experience with lupus patients, but I can tell you that the group of Inaki-sans in Emory has evaluated COVID patients in ICU, non-ICU, cured after the ICU time, and he has compared this double negative population that COVID patients are similar to what has been seen in lupus. He's a lupus guy. And some of the specificities that these people are making, in addition to anti-type 1 interferon and things like that, are signaling proteins involved in the response, in the antiviral response. Different because the virus induces a different specificity. But anyway, the processes that he has reported are very similar. Sans is his name, Inaki first name, from Emory. And you can find some interesting information specific for lupus because he's a lupus person.
Great. The other questions are more simple. One of them is related to the double negative B cells. By the expression of TBET as a transcription factor and CXCR3, it seems that it behaves like a Th1 cell, like the producer of interferon gamma.
The answer is yes, because TBET is a transcription factor also for autoimmune T cells. So when you have autoimmune T, you can find the same expression. I don't know CXCR3, but TBET for sure is a transcription factor for B and T cell autoimmune, and also CD8 autoimmune.
Okay, and the last question is related about statins, because we have some experience in the literature about the inhibition of activation in vitro when you pre-incubate endothelial cells with statins, even after challenging with toll-like receptor ligands like LPS. For example, they produce less pro-inflammatory cytokines. So would that be a good idea to try those B cells in vitro to inhibit them before with statins?
Well, yes, in vitro you can do it. It depends from the frequency because they go up with diseases. Sometimes I showed you from the sorting of the double negative from the stromal vascular fraction. We have very few from the blood, and in the blood they are already expanded. So yes, you can do this. You can use the micro wells in order to put them together. But the limitation is how clean they come from the sort. Because if you have a minimum contamination with switch memory or IgM memory, you can have an opposite result. But it's doable.
Thank you.
Questions? And after Tiago, if someone online would like to send a message, I'll check on the chat.
Thank you. Nice presentation, interesting data. It's good our presentation is not right before the lunch. And in fact, I have one question. It's a curiosity, and two, in fact, questions. The first one is BMI is not a good index, and liver steatosis correlates with this process, or it's a later index. Okay. And the second one is about the that result with B cells in vitro. The inhibitors you used indicates a lot of process, autophagy, oxidative stress, and metformin had a lot of effects, I don't know, inflammation. And do you know what came first, or the kinetics of this, the occurrence of this process?
Asking about which effect comes first and which is the pathway that is first inhibited. No, I haven't done these experiments because you need specific inhibitors of the different pathways. Not even in mice I have done these experiments. In humans, I think it's impossible. You need to have some conditional knockout mouse, and you can block one of the pathways of activation in order to know what comes first. No, I don't have the answer. It's difficult.
Because you have the beginning, the beginning picture and the end picture in the. So, thank you.
Thank you.
Thank you, Daniela, very much. I was looking at the computer, see if there was any question on online people, but they are shy. So we go to the, thank you very much. Was very nice presentation, Daniela. I will ask now Professor Maísa to come here, please. It's very nice to have Professor Maísa Semborolo here, because she's working for too long with other people after 80, and she's going to share some data with us. Thank you, Maísa.
So thank you for all of you to come and to share the data that we have been collecting. And thank you so much for the invitation. I'm very glad to be here and to have a chance to discuss a little bit with you all the things that we have been working with. And I'm going to talk about some clinical aspects. And we know that after the COVID pandemic, there was a decrease in the expectation of life in the United States. We can see at birth, there was a decrease in different groups of people, and also after 65 years old. And in Brazil also, we had about 2 years of decrease in the expectation of life. And depending of the vaccination, this decrease can be worse. But besides this, we still can see that Brazilians are getting old, and especially people that are 8 years or older increase in a faster velocity. And to build a society for all ages, the decade of health aging in the Americas give the directions that we have to support country planning and action. We have collecting better global data and promote research on health aging. Align health system with the needs of the older people, lay the foundations and guarantee the necessary human resources for integrated long-term care, run a global campaign to combat ageism, and improve the global network of age-friendly cities and communities. So all the countries are working on it, and a lot of countries had already a success in that. One of the examples of this is Ireland. That's considered one of the countries that is very good to live and to age. In Brazil, we have also blue zones. You can see in the map, in the blue color, places where you can find centenarians. You can find those in the northeast of Brazil, here, in the south and in the north. So all over the country, there are places where you can find these centenarians that are healthy people like those that are here in the pictures. This one is from Bahia, that one from Amazon, and this one from the south of Brazil is one of the city, Veranópolis, that you have a lot of centenarians that are very healthy. And it's interesting because they have different conditions of environment, but they are still independent and can work productively for the society. When we think about how to achieve exceptional human longevity, you have to consider that there are a lot of influence of geography, culture, and you have some identified points that can that you can have different combinations of them to achieve the exceptional human longevity. It's important genes are important, resiliency, environment, and chance, just to have a possibility to be healthy when you get old. When you see the life story in blue, you have conditions in this life story to construct the resiliency. That depends, the health depends of the social capital, depends of the property, education, income. So all these kind of variables are important to construct the resiliency that are going to give at the end of the life the possibility to be more adapted and to really have conditions to pass through difficulties that can present, especially in moments of crisis. So all this condition is important to be constructed during all the life, not something that you can get only when you are old, but you have to construct it during all your life. Because of all these things, and we have been worried about how can we treat or give good assistance for the elderly patients, we decide to look for what's going on with the elderly people that are 80 years and older, because they specifically are survivors. They show us that they have a specific way and response answers are not allowed, are not present in the scientific publications. So we decide to follow them in 2010 to see how we could understand a little bit better, to take care of them better. So we tried to select people that were 8 years and older from community, and we invited them by TV, newspapers, radio, and personal contact. And they should be independent for daily living. They should not have compromise or dementia or problematic disease. They should have possibility to answer the questions and to make the tests. And they should be very similar to the ones that live independent, sometimes alone, without any help, and they can take care by themselves without problem. So we selected 273 people, and we started to follow them. Every year we made global assessment and blood samples, and after about 10 years, we had 75 deaths, and some of them had outcomes like dementia, physical dependency or sensory dependency, or they have cardiovascular disease or cancer. But what is interesting is 127 were still independent without these outcomes. So selecting the oldest old who have compensated chronic disease and are independent may represent the aging that we assume is what most want to have. So we cannot be free of disease. Probably most of the chronic diseases are going to be with us as soon as we get older. But if they don't compromise the independency, the autonomy, it's okay. We can have a good life. So who are they? They were 291 women, much more than male, and you see. as we expected, because in most of the populations, including the different cities of the world, you have much more women than men when you study this age. The age was 85 plus minus 5. They have schooling 4 years, about 4 years. The mini-mental was 25, and the GDS was 3, the geriatric depression scale that you can detect depression symptoms. And the mental status, the marital status was, most of them were widow, and 20% were living alone. The religion more frequent was Catholic, and the profession, most of them were production of goods and services, and in home. And the skin color more frequent was white. It was by research. The color was done by research, not by the individual. When you see, most of them were from Brazil, and as much as her parents and grandparents. So, but we have a lot of people from Europe too. And the most frequent countries that we saw was Italy, Portugal, Japan, and Spain. Wow. So in order to understand how much is important the kidney function, first of all, we start to look for a way to better represent the glomerular filtration rate. So we did an Ioxol, that's a gold standard, and compare with 6 different formulas to estimate the glomerular filtration rate. And we found that these 2 that include creatine and cystatin are those that have a better accuracy. So we choose one of them to study the kidney function in this population. And when we try to see the functionality of this, because there are a lot of papers in the literature that shows that glomerular filtration rate, when decrease, compromise the functionality. And also there was a lot of papers talking about the vitamin D influence in the functional capacity. So we were interested to see if there was an interaction between vitamin D and glomerular filtration rate. And so when we studied that, we saw that they were independent. Decreased renal function associated with age compromises the ability to perform activities for independent life in the community. But we did not observe influence in specific domains of cognition and physical performance. So there was no influence in cognition, at least at that moment, and we could not find any interaction with vitamin D. On the other hand, low serum levels of vitamin D appears to be a marker of great risk of functional decline. So both seems to be important, and have an impact in functional capacity of the elderly, but they are independent for that. We also try to see how much vitamin D could influence physical tests. And we found that high vitamin D, I think I'm gonna close to, good for me, okay. High vitamin D, inadequacy, prevalence in those Brazilian community, the dwellers old, positively are associated with bone mass and dynamic balance, and negatively with PTH and osteoporosis diagnosis. So we already know about the osteoporosis diagnosis. We already know that they have an influence in the muscle activity, but at that time was something very important to understand, because we didn't have too much information about this. And we saw that vitamin D more than 20 nanograms by ml was associated with better bone mass and lower PTH levels, make them much more specific, that we have to maintain those elderly people with levels above 20 milligrams by deciliter. That's something that we now try to obey. And we didn't find any correlation with muscle mass or mass in this population. When we saw the FGF-23 and functional capacity, in this population, this marker was associated with comorbidities related to age and renal function, but not to functional capacity. So FGF-23 in this population, we have the lowest tercile and to estimate GFR. That's something that we already know, that there is a strong correlation between FGF-23 with the kidney function, but we could not find any correlation with functional capacity. And you see here, the only thing was with cystatin C that represents the kidney function. So when we compare centenarians and what some publications show us, we see that we have common age-associated disease, except cognitive impairment in centenarians in general. Before 8 years old are those that we can call survivors, because they have age-associated disease before this age. And we found, most of the authors found 24 of males and 43 of females with this phenotype. And those that no experience age-associated disease before 8 years of age are called delayers, 43% both males and females. And those that no experience age-associated disease or escapers, 15% of males, 30% of males. You can note that the survivors, you have much more females. And the escapers, you have much more males. It looks like that men that can survive until a very old age are in better conditions than women. Not so many survive, but those that survive look like they are with less common age-associated disease. In our study, when we look for cardiovascular disease, stroke, and cancer, that are the diseases that kill more, we found 90% of them were survivors, 21% delayers, and 60% escapers. So most of these people don't have cardiovascular disease, stroke, and cancer at age 8 years and old that we invited to follow. When we saw the common age-associated disease as cardiovascular disease, stroke, cancer, hypertension, Parkinson, COPD, diabetes, thyroid disease, osteoporosis, we saw that most of them were survivors, and some of them 15% delayers, and only 10% escapers. So you age with chronic diseases, but does not mean that these chronic diseases are going to be something that make you be dependent or have a worse life. So after that, we decide to look for how some variables like age, sex, skin colors, home country, civil status, living alone, religion, profession, schooling, mini mental state examination, geriatric depression scales and morbidities were correlated with the outcomes that we saw in these patients. And what we saw was that long-lived elderly classified as delayers and escapers, those that have the common age disease after 8 years old, have a subdistribution of risk of death 3.2 times greater than that of long-lived elderly classified as survivors. So you see here that those that are delayers and escapers need more attention. If they are until 8 years old with chronic disease, maybe the body is in some way capable to control the conditions of the factors that are involved in this chronic disease. But after 8 years old, if they start to have common chronic disease, it means that there is imbalance and you have to look for what's going on because they have a high risk of death if you compare with the others. So maybe we have to give more attention for this group, different from the others. When you see the outcomes that happen, we can note that 46.5% didn't have outcome after 10 years. And dementia was the second one more frequent. And 13.6% died without any time of dependency. That's very interesting. And cancer and cardiovascular disease was not so common as an outcome, but they were very important because when they happen, they cause death in this population. So most of them die, and these are really the disease that kill more, and you have to have a lot of attention, as we do for the younger population. So cardiovascular disease is still a big challenge in this population after 8 years and old. The infection especially was related with the geriatric depression scale. And 1 unit in geriatric depression scale leads to a 19 increase in the subdistribution of risk of death from infection. So depression seems to be correlated with the possibility to have infection. Maybe because the people that are depressed or have symptoms of depression stay more in bed or don't move too much, don't make exercise, are not stimulated to do things. I don't know, but it's important to pay attention of that. And we also observed that there was an inverse association of depressive symptoms with vitamin D levels. When we saw the vitamin levels, especially those that had less than 20 nanograms by ml, they had more association with depression. It seems that vitamin B, when in balance, can contribute to a lot of problems, especially in this age population. About the immune profile and body composition, we found that in independent oldest old, ERP, the relation between CD4 and CD8, proved low, but we have a high sensitivity of CRP that was associated with body composition. And in fact, it's something, but it's still more interesting that the CRP in this population, the level of that was very low. They were not inflamed. They had not any alert to show us that they could be with this kind of problem. Probably they have a different metabolism of the cellular cells that can contribute to that, but the inflammation was not detected by the level of CRP. Besides, the CRP was correlated with the obesity in this population. We know that there are a lot of age-related change in cardiovascular system. There is an increase in central aortic stiffness attributable to increased collagen cross-linking and elastin fiber degeneration. There is an altered left ventricular diastolic relaxation and increased myocardial stiffness, decreased in responsiveness to beta-adrenergic stimulation, impaired endothelium-mediated vasodilation, altered balance between intrinsic thrombosis and fibrinolysis, chronic low-grade inflammation that we already discussed. And all these conditions have implications to acute coronary syndrome, especially increased atherogenics and geriatric syndromes, including frailty in this population. So we saw that the 1 year increase in age led to a 22% increase in the subdistribution of risk of dying from cardiovascular disease. So we see here that there are really an influence of age that's expected because of those alterations that are done related to age. And the layers and escapers have a subdistribution of risk of death from cardiovascular disease 2.2 times greater than the oldest old classified as survivor. So again, no, those have more chance to have the outcome. Those, the layers and escapers, have more chance to die. And so we have to pay a lot of attention for those that get sick after 8 years and older. And when we see the association of the corporal composition and the stiffness, arterial stiffness, we see that in long-lived people, body fat percentage was directly associated with arterial stiffness and inversely associated with the amount of LM. These findings may be associated with increased cardiovascular risk in this population. So they were considered healthy, no, at least in terms to be independent, don't have problems with daily activities, have common chronic diseases compensated, but they have difficulties and changes that can increase the mortality in the possibility to have outcomes in this population. And we also saw that increased levels of nitric oxide may represent a marker or better health in longevity outcomes. So those that were older in this population, they have increased levels of nitric oxide. And the combination of glomerular filtration rate greater than 45, HDL greater than 60, and vitamin D greater than 20, and CRP and homocysteine at the limits lower reference values, defined a more favorable profile associated with a lower risk of dementia, stroke, and death in this population. So when we saw some risk factors that contribute to increase the chance to have cardiovascular disease or death by cardiovascular disease, we saw that the there is a cluster of these elderly that have more favorable profile of these factors. And to look for the outcome of dementia, first of all, we try to see if the mini mental examination exam was a good exam to detect dementia in this population. And we found that the score of mini mental state examination, corrected by schooling, of course, and in these patients with fever chronic disease were associated with a higher incidence of dementia. So this instrument was good to detect dementia in this population, besides the age. And when we saw the mortality, we saw that an increase of 1 unit in the mini mental examination led to a 12% reduction in the subdistribution of risk of death and other causes and unknown causes. So to be cognitively healthy, improve the chance not to die. So it's important to preserve the cognition, because when the cognition is decreased, the chance to die increase. Again, when we compare centenarians in the literature, 25% are cognitively intact. Delayed clinical onset of impairment until the average age of 92 years. Cancer is diagnosed at later age. Vascular disease, relatively late in life. In longevos that we saw, we observed no difference between the sexes. So in this population, there was no difference between them. Both the incidence of cancer and mortality from this cause were not associated with any of the variables studied, not even age and gender. It's interesting. We were not expected that. The events that had a higher incidence with increasing age were death as the first cause and death from cardiovascular disease. The long-lived, the layers 10, those that have common age disease, and escapers, 10 common age disease, had a higher risk of deaths from cardiovascular disease, from other death, and from unknown causes. And mortality from infection was greater the number of depressive symptoms. So this is maybe one of the points that must be very interesting to focus. Some of them, the oldest old, died without experience any of the events studied, without having to go through a period of dependency. So that's something that most of the elderly that you ask say that they don't want to be dependent. And maybe if we can control most of the variables that can give bad outcomes and give them a chance not be dependent, it's something that is going to help them to have a better life. So these are all the team that have been working on this data, and I always have to say thank you for everybody. And I finish with the phrase that the survival of independent, long-lived elderly is the greatest expression of development to be conquered. Thank you for your attention.
Thank you, Maisa, very much for this nice presentation and for sharing your data with us. So, questions, please.
Thank you very much. I enjoyed a lot your presentation. And I have a question for vitamin D. So are the levels of vitamin D that you are showing and that you measure in serum plasma samples are following a supplement with vitamin D, or are these just steady state levels?
Steady state levels.
And how much these levels of vitamin D are correlated, if you know, with the immune function of these individuals? Because there is a very well-known effect of vitamin D on fat mass, and you have shown some of these relationships. How is the effect on immunity? I mean, infections, protection from infections, etc. Do you have any results on these people that have at least a protective level of vitamin D in serum?
Oh, thank you for your question. No, I still don't have it. I'm looking for to analyze the data, including vitamin D, and see if there is any correlation. The last study that we did about immune system was published this year. So we are now going to start to review the data during the 10 years and see if the variation of vitamin D could have some influence in the immune system in this population. I think it's a very interesting point to see.
Can I say something else? We have a study with cardiovascular people, and these are, we have a number of individuals with type 2 diabetes and obesity that are diagnosed with mild to severe cardiovascular issues, and they have to undergo CAB surgery. These people have a very thick layers of epicardial fat in the periatrial and pericoronary area of the heart. And in these people, the levels of vitamin D are almost undetectable. So they are put on very high levels of supplementation with vitamin D just to help them to counteract the effect of the deposition of fat.
Oh, thank you for the information. In fact, I believe that vitamin D is very important for these elderly people. I think the imbalance of vitamin D in them is responsible for a lot of dysfunction that happen. I don't know how much we can improve, but I'm sure that vitamin has is guilt in this kind of patient. I'm sure about that.
Thank you.
Thank you.
I just like to do a compliment on the question you asked, Daniela, because I have done vitamin D and tried to correlate with some immunological profiles like naivety cells, CD4, CD8, B cells, CRP. But as I had such a small number of healthy, was from 80 to 100 individuals, was a very small number, so I couldn't find any correlation. But I am sure that if you do more numbers, you can find something. And especially, I could not talk to the individuals, so I don't know if the only one that have more than 30 milligrams of vitamin D on circulation was just one person, one man, and I couldn't know if he was taking supplements or not. So I think Maisa will be able to interview them and have this answer because I couldn't. So, but for sure, vitamin D will make a huge difference on their lives. But there is one problem here. We are used to not take sun because of all the skin cancer history. So even younger people, when I did the comparison between 30 years old people and these 80 plus years old, was just the same actually. The youngers also don't have good levels of vitamin D. And there is something else we don't know, at least I don't know, let's correct it. I don't know if the absorption of vitamin D by the intestines is the same. Yeah, so there are some points we have to think about. But I think, again, it's important to have, at least at old age, to have a good level of vitamin D for not just for the immune system, but for everything, bones and so on.
In fact, we did a study in the SESC community. That's an institution that you practice exercise. And there were 430 elderly women that were practicing exercise there. And what we did was we made physical tests in one day, and then we supplemented them with vitamin D during 1 year. And after we repeat the blood samples, the DEXA, and the physical tests. And they were elderly that were practicing exercise for at least more than 1 year, because in that institution, if you don't go, you are out. You have to go very straight, otherwise you cannot stay there. So they were working very hard during more than 1 year, some of them for 10 years at least. And when we give them vitamin D, they improve the physical tests. It means that if they don't have a good level of vitamin D, maybe your performance is not so good as could be if you have good levels. And so that I believe that vitamin D is important. But those elderly women were not 8 years and older. They were younger. The mean age was 72, something like that. But if it's important to them, maybe it's even more important to elderly people 8 years or older.
Questions?
Okay, just to complement what you think about the absorption of vitamin D, most of these elderly people in the outpatient service are under vitamin D supplementation, and they have more than 40 nanograms per ml. So they can reach more than 20 after supplementation. So I don't think that absorption is a problem. Maisa can tell us something else about this. These are able to absorb vitamin D if they are under supplementation.
Well, when you give them supplementation, they are okay. They can absorb.
Yeah.
We didn't saw any problem with that.
Thank you for your presentation. It was very important to me.
Thank you.
Do you have any studies on the microbiota of these older individuals?
Not yet. There are a lot of postgraduate students that are interested in that. We believe that maybe we are going to start to doing that. We are waiting for financial support from the institution to see if we can make this work. I think for this population, maybe it's very important. And I note that Daniela said a lot of times that microbiota was involved in a lot of situations. And I believe that's something that we must pay attention.
Thank you.
Thank you.
Even because it's very different the composition of the microbiota between healthy elderly, nonagenarians, and centenarians. So they're all clustered very
That's true.
and far away from the young and the middle age. So there should be something that is like very interesting studies because you have two cohorts very well defined.
Thank you.
And for sure, when you be able to do this microbiome study, you're going to have different profiles of individuals, maybe different from what you presented here today, because as further you go investigating small pieces, further you can distribute them on different groups. That's my feeling.
Just to know your opinion about this, Daniela. In the centenary studies, the microbiota is completely different from other ages or even in older people. But one thing that we are thinking that is better than study the microbiota, that is a very complex study, is to detect the metabolic derivatives from this microbiota. Mm-hmm.
These studies are extremely expensive. So you have to either do a targeted metabolic analysis of serum samples, or if you do everything, it's something that is absolutely difficult, even within the definition of the hierarchical clustering of the different. I think that the metabolites are even more important because they are reflecting everything that is also coming from the macros.
Yeah.
So, if I have to favor one of the studies right now,
Yes, we are thinking about short chain fatty acids and peak result, for example.
this can be done in thousands of hours.
Mm-hmm.
That's what we're going to do.
It requires a subsequent. So plasma, you have so many data, interesting data. Non-plasma samples is about expensive.
Useful.
Thank you.
Thank you very much.
Now, I have a great pleasure to call here Professor Thiago Rodrigues from Federal University of ABC. He's working on mitochondria, and I think he has very good things to tell us. Maybe more questions than answers, but very interesting anyway. Thank you very much for being here.
So good morning. I'd like to thank Valkyria for this kind invitation to be here. It's a pleasure to be here. And I'd like to congratulate you for this symposium. In fact, I'm out of this area. I came from the biochemistry, and I have studied mitochondria for years, but in a different context. In my lab, we studied cancer, mitochondria, and cancer, and everyone who works with cancer already see this picture, the hallmarks of cancer by Hanahan and Weinberg, and our research interests are related to these conditions, the energy metabolism and resistant to cell death by cancer cells. So I tried to do my best to collect information about mitochondria and aging. I put this information together. Maybe the slides are very crowded, but I try to bring this information to generate the discussion. Mitochondria are considered the powerhouse of the cell for years. This is not the current view, but I think this view is because mitochondria have in the inner membrane the proteins able to transport electrons and generate a proton electrochemical gradient. And this gradient is used to release the ATP synthesized in the ATP synthase. And this is the major way to generate ATP inside the cells. More than 90% of the ATP inside the cells are generated by the oxidative phosphorylation under aerobic conditions. So this is a classical picture for the Leninger's book, and the energy metabolism collects the energy from all the macromolecules, amino acids, fatty acids, and glucose into acetyl-CoA that can be converted to carbon dioxide and water in the Krebs cycle. And these reduced coenzymes donate these electrons to the respiratory complexes, and the electron transport is associated with proton transfer from the mitochondrial matrix to the intermembrane space. And this membrane, the inner mitochondrial membrane, is a special membrane, very special, because it's composed by 70% of protein and 30% by lipids. These lipids are, there is a special lipid, the cardiolipin, in this membrane. So it's a proteic membrane, not a lipid bilayer. So the mitochondrial inner membrane is impermeable to all the ions and solutes, and this electrochemical gradient generated by protons, it's able to release the ATP, as I said. But mitochondria has other functions inside the cells than generate ATP. In 1979, this is the first mentioned to this process named the mitochondrial permeability transition. This process occurs in mitochondria inside the cells, a transitory permeability. And this permeabilization is associated to release of some factors that trigger cell death. So the next 20 years here was dedicated in the field to discover what are the proteins involved with the formation of this spore. And nowadays, we believe that the adenine nucleotide translocase and this porine VDAC makes a megapore between the mitochondrial membranes, and with the participation of cyclophilin D, it's a calcium-dependent process, and when this assembly occurs, mitochondria can suffer osmotic swelling, the dissipation of the mitochondrial transmembrane potential, release of calcium, and pro-apoptotic factors. And then in the 80s and 90s, the MPT was very studied by researchers in the area and associated with pathological conditions and disease. After this, in 2000s, another type of permeabilization was described, named the mitochondrial outer membrane permeabilization. This process is caused by a pore formed by Beck's proteins that are translocated from the cytosol to the mitochondria and oligomerize in mitochondria outer membrane. In this pore allows the release of cytochrome C to induce apoptosis. So there are two types of permeabilization described for mitochondria, and this permeabilization is associated with cell death. And then this is studied in several types of models, including diabetes, inflammation, infection, chemicals, and cancer, and induced by drugs. And this process are studied in the field. And then, so independent of the process, the mitochondrial permeabilization can be caused by these situations and calcium overload. Mitochondria participates of the calcium, cellular calcium homeostasis, but calcium overload can trigger the mitochondrial permeabilization. Also, the free radicals, reactive oxygen and nitrogen species can damage mitochondrial macromolecules and induce this permeabilization. Drugs and diseases or pathological conditions can trigger this process, and this results in mitochondrial swelling, the mitochondrial transmembrane potential dissipation, calcium release, thiol groups of proteins oxidation, and release apoptotic factors. So I'm saying this because all of this can be measured as indicative of mitochondrial permeabilization in isolated mitochondria and also in intact cells. As an example, this is a study from my lab we used isolated liver mitochondria as a model to study a drug-induced mitochondrial permeabilization. So it's just an example. We show that this drug can reach the mitochondria, oxidize thiol groups, and in the presence of calcium, there is an opening of the permeability transition pore, leading to the mitochondrial swelling, dissipation of the transmembrane potential, calcium releasing, and cytochrome C releasing. This is an illustrative figure, and we reach this data, and we propose this scheme based on the use of inhibitor of this process. Calcium chelators can inhibit this process. Competitors of calcium, cyclosporin A is an immunosuppressive agent, but can bind to the cyclophilin D, and besides an immunosuppressive agent, cyclosporin A is a classical PTP inhibitor, as described as a classical inhibitor of the permeability transition pore. We described the molecular mechanism of mitochondrial permeabilization that's induced by this drug. And then we start to investigate if this occurs in cells. We use these hepatocarcinoma cells, and first we perform the concentration response curves to determine the cytotoxicity of this drug. And this drug is an antipsychotic agent, and it's the most cytotoxic derivative with the EC50 for cytotoxicity, about 45 micromolar. So the question is if this cytotoxicity is associated to the effect on mitochondria. And then we measure the mitochondrial membrane potential inside the cells, and we vary the drug concentration. And then we plotted the drug concentration with the dissipation of the mitochondrial membrane potential, and we found the same EC50 value. And by suggestion of the reviewer, once I submit this paper, they asked to join this information with mitochondrial swelling in isolated mitochondria from this published paper. We plotted the drug concentration and mitochondrial swelling, and we found the same EC50, suggesting that the mitochondrial permeabilization is involved with the cell death induced by this drug. So this is an example on how we can study mitochondrial function in isolated mitochondria and then also intact and whole cells. But talking about aging, I have to study a lot to understand a little about aging. It's not my field. And I put some considerations here. It's my considerations based on what I have read during this time. And I saw that the cellular and molecular mechanisms responsible for aging are still poorly understood or not completely understood. This is my opinion, but I think there are a gap here. And then I tried to define aging. And this was a problem inside my head, because I found some definition like aging is a physiological this question mark is my question mark, if aging is really physiological or when associated with another disease is pathological. I don't know. I want to discuss this with you. Leading to a general decline of physical and mental capacities. So it is too open, but regarding the molecular aspects, aging has been associated with cumulative cellular lesions, leading to a progressively to organ dysfunction and death. And then reactive oxygen species seems to be also involved with the pathogenesis of aging. I don't know if this is true all the times, but several papers bring measurements of production of free radicals and quantify the damage promoted by these free radicals in macromolecules, lipids, proteins, DNA, to associate with this with aging. And then the mitochondria as the main source of cellular ROS, because during the electron transport by the respiratory chain, part of this electrons escape and promote an incomplete reduction of the oxygen, not generating water, but superoxide anions that can be converted to hydrogen peroxide in the presence of metals, hydroxyl radicals prone to attack the macromolecules. So I saw a lot of review papers with mitochondria, oxidants and aging, mitochondria and longevity, mitochondria and aging. All of them put ROS in this battlefield. So, and then I try to find some data, not mine data because I don't work with this until now, but I found some interesting data. This one was published by a colleague here in Brazil, Professor Alicia Kowaltowski from USP, studying caloric restriction or uncoupling of the oxidative phosphorylation, and she showed that increased lifespan in Saccharomyces cerevisiae. And the model was the caloric restriction model in yeast was to high glucose in the media and low glucose in the media. So in this situation, the caloric restriction, there are a higher oxygen consumption and a lower ROS production. And this can be seen by this ratio. And so in caloric restriction, there is more oxygen consumed and less ROS production. So it indicates that mitochondria, the electron flux are rapid. Some mitochondria seems to be uncoupled, and this decrease the electron escape, decreasing so the hydrogen peroxide formation. And this can be seen here by a kinetic measurement of hydrogen peroxide. This is the basal levels, 0.14. And then when we add an inhibitor of the respiratory chain, this level increase significantly. And in the presence of an uncoupler of the oxidative phosphorylation, there is a decrease in ROS production. So then a viability experiment was performed to assess the lifespan. And we have here the control. And in the presence of another uncoupled D-nitrophenol, uncouple of an uncoupling agent of the oxidative phosphorylation, there is an increase in lifespan in yeast. And then this was also demonstrated by the same group in mice. And here with this uncoupler, D-nitrophenol, and increasing the oxygen consumption in brain and liver, and also decreasing the ROS production in these tissues, and also increasing the lifespan of these mice. Then I found another paper comparing the outcomes of caloric restriction in young and old rats. And then you can see here the ATP production in these groups. We have here the control group, young. This is the old group. ATP is lowered by a half. And then in the this old group plus caloric restriction, this capacity of ATP synthesis is restored. So it's a descriptive data. And then this issue has been reviewed in the literature, but it seems a consensus that there is mitochondrial alterations in aging. And these alterations is, it is not positive, it's mitochondrial dysfunctions. So in this review, there are the original articles showing that there is a ATP synthase disassembly, the loss of the crystal structure, the mitochondrial membrane potential is lower in aged cells than young cells. There is a calcium overload, and then a higher probability to the opening of the mitochondrial permeability transition pore. So here, it was used isolated brain mitochondria from rats to test the occurrence of the mitochondrial permeability transition in animals fed ad libitum or with caloric restriction. So here is the extra, it's not cytosolic because it's isolated mitochondria, but it's an extra mitochondrial calcium measured with calcium, the dye calcium green 5N. And here we have calcium addition, small amounts of calcium addition. And when calcium is added, there is an increase of mitochondrial, extra mitochondrial calcium, and mitochondria do the uptake of this calcium. And then another addition, increase extra mitochondrially, and then mitochondria do this uptake again. So what is tested here is the calcium retention capacity. as an indicator of the mitochondrial permeability transition, because when mitochondria is permeabilized, calcium is effluxed from the mitochondria. So in red line is the animal feed ad libitum, and this blue line with caloric restriction, 40% of caloric restriction. And then these animals with caloric restriction has a high calcium retention capacity. And as I told, this calcium is effluxed probably by the mitochondrial permeability transition. And then they used cyclosporin A as an inhibitor of this pore, and then no difference between these groups, showing that the caloric restriction is associated with increased lifespan, and it's modulating the mitochondrial permeability transition. So maybe there's a link between this process. Well, let's see my time. Besides this function of mitochondria to produce energy through the oxidative phosphorylation, mitochondria exert a lot of other functions. I try to summarize some of them here. Mitochondria is a metabolic hub inside the cells because several metabolic pathways occurs inside the mitochondria, including the TCA cycle, urea cycle, the oxidation of fat acids, amino acids, heme group. As I told you, mitochondria can produce free radicals, and because of this, mitochondria has a complex antioxidant system. Mitochondria also participates from the calcium homeostasis, control metabolism, and this crosstalk between mitochondria and the endoplasmic reticulum can also contribute to the calcium homeostasis. It was described that there is contact sites between mitochondria and ER. Mitochondria can suffer the permeabilization, and this is related to cell death. And this is controlled by BCL2 family proteins, a family of proteins composed by anti-apoptotic proteins and pro-apoptotic proteins, controlling mitochondrial permeabilization, but also calcium homeostasis and also metabolism. These functions are being discovered along the time. And then I'd like to add the description of another functions here. Mitochondria has a tightly regulated process of quality control. It's a kind of autophagy. It's a specialized autophagy named mitophagy, where damaged mitochondria or old mitochondria are eliminated by autophagosomes via autophagy. And then, at last, I'd like to highlight this process named mitochondrial dynamics, where mitochondria can suffer this fusion and fission process, and also the synthesis of the new mitochondria named mitochondrial biogenesis. This is a complex process named mitochondrial dynamics. This is the hot topic about mitochondria nowadays. And then I try to show some data about this point here. So metabolic reprogramming, it's not a new field of study. Otto Warburg have studied this since 1926. And then when he was studying the metabolism or the acidification, extracellular acidification in cancer cells, and he described that cancer cells can use the glucose, transform the glucose into lactate, even in the presence of oxygen. This is named aerobic glycolysis. And this was named as Warburg effect for cancer cells. But now you know that other cells than cancer cells can suffer this Warburg effect, changing this metabolism to adapt to the new situation. And then mitochondria is involved in this process through the mitochondrial dynamics. So mitochondrial dynamics is a highly dynamic process coordinated fusion and fission cycles. In this process, a mitochondria can suffer a fission process. In this process, there are some proteins already described. The main one is DRP1, NFF, and FIS1. In this fission process, DRP1 can be phosphorylated and then activated. And since DRP1 is activated, it's translocated to the mitochondria and oligomerizing, promoting a strangle ring here, and mitochondria can be divided in two. This process is named fission, and is not a random process. It's controlled by proteins. And this smaller mitochondria, on the other hand, can fusion. And this fusion process is coordinated by these proteins, mitophysin 1 and 2, and also OPA1 that is involved in the crystal, the new crystal formation. And then this occurs all the time inside the cells, according to energy demand or the metabolic state or the presence of a disease. So this is a balanced process that can be altered in a physiological condition to adapt to a metabolic state, or in the presence of some pathological conditions. Drugs, pathological conditions include aging. Here is images of mitochondria. Here is stained mitotracker green, and these cells were imaged by confocal microscopy. Just to show the influence of these proteins I mentioned in this process. So this is a control cell, it's basal condition. Mitochondria has this tubular phenotype. And when the mitophysin fusion 1 was decreased, knockdown of mitophysin 1, it's a protein involved with the fusion process, and then fusion was decreased, mitochondria is smaller, very smaller. This is a fragmented pattern. On the other hand, when the DRP1, the protein involved with fission, was knocked down, mitochondria appear as a hyperfused phenotype pattern. So we can evaluate the mitochondria or the mitochondrial morphology by using confocal microscopy or even electronic transmission electronic microscopy. And then I add here the mitochondrial biogenesis is a process controlled by PGC1 alpha and this transcription factor TFAN generating new mitochondria. And then here, this mitochondria can instead participate of this fusion process, can be eliminated by mitophagy, as I told you. Also, a damaged mitochondria can release the apoptogenic factors and cells undergoes to the apoptosis. So this is a simplified general view of the mitochondrial dynamic process. So the question is, the modulation of mitochondrial dynamics is able to ameliorate pathological conditions or diseases. I insert here some considerations. There are a few chemical tools to investigate the mitochondrial dynamics in biological models. There are still a few drugs to inhibitors or stimulators of this process to investigate this. The available tools like MDIV1 is one of the most known inhibitors. It's an inhibitor of GRP1, have off-target effects already described. MDIV1 can inhibit the complex one of the mitochondrial respiratory chain. And then genetic manipulations, is the knockout, knockdown, or overexpression, have provided useful insights in this field. But thinking about treatment, the possibility of the treatment of the patients, and we have this drug development phase to overcome. We have molecules, and so we need to test new and older compounds able to modulate the mitochondrial dynamics. Again, I will present some data, not my data, but data about mitochondrial dynamic manipulations in some models. The first model, it's a heart failure model. I consider a very nice data. It was recently published in Nature Communications. We have here a colleague from Brazil, Professor Júlio Batista from ICB USP. And they have this well-established model in rats with heart failure. So there is a electronic microscopy here with animals, normal animals shown. Here is the heart failure control. We can see mitochondria are smaller. There's a problem with this tissue. And they described a peptide. In fact, they screened several peptides, and they found one, this one, the better one, named Samba. It's a suggested name, so, yes. And Samba promoted mitochondrial fusion. And this preserved mitochondrial function and also prevented from the damage promoted by heart failure. So part of the data I highlighted here, we have an increase in the number of mitochondria. This reflects the fission process. We have more mitochondria, but smaller. More mitochondria and smaller. This is the size of mitochondria. And this peptide was able to restore the levels to the control levels. So the bioenergetic parameters, respiratory control are also restored, mitochondrial respiration rate in state 3 mitochondrial respiration rate, generation of ROS was prevented by this peptide. And then there are a lot of data, but they proposed a mechanism showing that PKC is activated during the heart failure, and the activation of PKC bound to the mitofusin 1 and block the mitochondrial fusion in this model. And mitochondrial fusion could rescue mitochondrial and energetic demand in the cell. And then there is mitochondrial fission, decreased mitochondrial bioenergetics, increased cell death, and a poor heart failure outcome. And then when the cells is treated with this peptide is able to bind to PKC. It's in fact acting as an inhibitor of the activation of PKC, and this allows that mitochondria become hyperfused, and this increase improve the heart failure outcome. So this is again an example on how to manipulate the mitochondrial dynamics in a model of disease. So I have another data here exemplifying this in cancer. This data is from our lab in a melanoma model. We used cells, the cultured cells in vitro. These cells have this mutation. It's most prevalent mutation in melanoma, BRAF mutated. And then we investigated the effect of this drug, that it's a target therapy, gold standard treatment of BRAF mutated melanoma. Vemurafenib from Roche pharmaceutical company. And vemurafenib was developed as a BRAF inhibitor. This drug inhibits the mutated BRAF and then blockade the downstream signaling of the MAPK signaling pathway. So we validate here the model showing that in vemurafenib treated cells, we have decreased MAC-ERK signaling in 12, 24, and 48 hours, decreasing the activated ERK. And the literature have shown that activated ERK is able to phosphorylate and activate DRP1. Is one of the activators of that protein involved with mitochondrial fission. And then we would like to know if this effect of vemurafenib has an impact on mitochondrial dynamics. So we treat these cells with vemurafenib, and we have a significant increase of the mitochondrial mass. You use MitoTracker green and confocal microscopy here. And we investigate the expression of the proteins involved with mitochondrial dynamics. And then, as expected, cells treated with vemurafenib have a decreased phosphorylation of DRP1. And then this fission process was inhibited. And we investigate also the fusion proteins, and there is a slight increase in mitofusion 1, 2, and OPA1, but the main effect seems to be on GRP1. And this is the phenotype. Electron microscopy, non-treated cells, smaller mitochondria. In cancer cells, the balance of mitochondrial dynamics seems to be to the fission. And when treated with vemurafenib, we have a beautiful elongated mitochondria. But we would like to, we don't know if this mitochondrial fusion promoted by vemurafenib could be a resistance mechanism against the drug. So we don't know this answer yet, but we try to affect mitochondria with another drug plus vemurafenib. We use the inhibitors of the respiratory chain, rotenone inhibitor of complex 1, and mycin inhibitor of complex 3, and the uncouplers, CCCP, and all of these substances enhanced the effect of vemurafenib. Here it's a slight effect. And then it seems that target mitochondria, I don't know if mitochondrial dynamics, we can improve the effect of vemurafenib. So another example about the manipulation of this mitochondrial dynamics can be used in diseases. So here the immune system. I'm not immunologist, so I teach on test here. I put as a simplified scheme to me to remember me about this process. If this is wrong, please, Valkyria, correct me. But I found in the literature robust data about mitochondrial dynamics in cytotoxic T cells. There are some groups working with this in the world, and I found several convinced data about mitochondrial dynamics alterations in these T cells. There are a lot of papers, and then I bring some summary figures in reviews papers. I chose 2, and it seems a consensus that the naive cells has a oxidative, it's an oxidative cell, I mean, obtain energy from the oxidative phosphorylation, and when these cells are activated, they become glycolytic cells. There are several experiments, most of them obtained by seahorse measurements of extracellular acidification and oxygen consumption rate. And these activated cells, when it becomes memory cells, these cells return to our oxidative metabolism. So there are a lot of data showing this. This review was published in 2010 by Professor Dirk Brenner from Denmark. And this one, it's another research in the area, Professor Erika Pierce from Germany. There is a lot of papers published by her group and others, shown that this naive T cell have basal metabolism, and when these cells are activated, the glycolysis, aerobic glycolysis is stimulated, and when these cells return to a memory T cells, is a switch to a oxidative metabolism, mitochondrial metabolism. So, and then her group showed that we have here elongated mitochondria, the fusion process. During the activation, we have fragmented mitochondria. Fission process was stimulated, and in memory T cells, we have more elongated mitochondria than here, the hyperfused mitochondria. So it seems a consensus that when the fission process was stimulated, this mitochondria, this cell has a glycolytic profile, metabolic profile, and when fusion process is stimulated, these cells have a glycolytic profile. But this is economy in terms of energy. So the production of ATP by glycolysis occurs, generates by test book, the yielding of 2 ATP. But we can keep in mind that we have alpha keto acids feeding the TCA, and the mitochondria is not completely off. It's related to the use of glucose to pyruvate enter into the mitochondria or transform into lactate. This process, the glycolysis, the obtention of energy by glycolysis is faster and very much faster, but the yielding is lower. So I think it's a balance. I don't have this answer, but I think it's a change of the steady state according to the demand. Yeah. In cancer cells, glycolysis is used for this another functions, including the phosphate pentosis, the pentose phosphate shunt, to generate antioxidants, and energy is provided by amino acids, the glutaminolysis. And then I don't know if this is occurring here. It's a kind of Warburg effect, and this work here by Professor Pierce, there is a specific paper showing that the oxidation of fat acid is increased to generate acetyl-CoA to feed the TCA cycle. He quantified the enzymes involved in fat acids oxidation pathway, and so it seems that these substrates is driving to the other pathways depending on the need. Okay.
So I know the word from Erika, and I'm asking if it's always the availability of the substrate generates the difference in the mitochondria in effector versus memory descent. Is it possible with specific inhibitors of fatty acid oxidation or oxidative phosphorylation to understand which is the major function rather than is that possible?
I think it is. It's the kind of thing I would do in my lab if I was working with this, because we can use inhibitors here, here, and also to select specific steps to knock down the enzyme and to isolate the one pathway and another, to understand the contribution of that pathway to the final process. And then about, I'm sorry, the time, I think it's okay. Thank you. And about aging, I try to find all the possible data about this, but I became frustrated because there are not so much works addressing the mitochondrial dynamics in aging. So I found someone, yes, of course. And then, so I'm presenting all the papers I found, I have found, I find, but maybe someone are out of here, but I try to find all of them. So in this paper, authors studied muscle, and they try to compare young and old. It's humans. The age here is a mean of 22 years old, and here 70 years old. And we have here twin individuals, half and half, male and female. And here also 24 individuals and half male and female. And they found decreased mitochondrial number per area of muscle, but there is not the image of the electron microscope to see this. It's just this bar graph, and then it's not possible to see if mitochondria is fission, is smaller or elongate. So, but they report a decrease of the number of mitochondria per area, and they evaluate this citrate synthase activity as a marker of mitochondrial mass. It's a common experiment. Use the activity of citrate synthase or mark with the mitotracker green fluorescence. And they found also a decrease in the citrate synthase activity suggestive of decreased mitochondrial mass. But I highlight 2 points here because they describe that these individuals have different levels of physical activity. And then this data was not corrected by the muscle mass. I don't know if I'm wrong, but older people can have less muscle mass. So I don't know if this is just a reflect of the amount of muscle or if there is a molecular alteration. So this is one study I found. Another one, also in muscle, and this is rats. Human. If I understood well, this is a biopsy from knees during the surgery, and to have a small piece of muscle, and they did this experiment of tunnel staining to show apoptotic cells varying with aging, and there is an increase of apoptotic cells in muscle with the increase of aging. And they found that an increased expression of apoptosis induction factor. It's a protein released by mitochondria to activate caspase, to activate DNA fragmentation independent of caspases. And then also the caspase 3, if it's a effector caspase, and they found an increase of AEF with age, but not caspase. And then they conclude that caspase-independent apoptosis increases with age in muscle. But these are the experiments, are just descriptive. And here in Saccharomyces cerevisiae, we have this is an interest model because it's easy to manipulate the genes. And then these authors have a wild type yeast here. The knockout of this gene that is correlated with DRP1 in mammals. MGM1 is like OPA1 in mammals, so fission, fusion, and both. And then they showed that the knockout of DRP1, its expected mitochondrial becomes elongated, has an increase in the lifespan, but the knockout of OPA1, it's related to the crystal formation, so it's expected that decreased lifespan. But the double knockout have a decreased, a higher decrease in the lifespan. So just this descriptive data also. And then I found another paper. This is with more data, but it's a model to investigate the mitochondrial quality control. They used this model of Drosophila melanogaster, and they overexpressed parkin, that protein involved with mitophagy. And authors showed that an increase in the lifespan with parkin overexpression, and this is associated with increased activity of the citrate synthase, showing the increased mitochondrial mass, and also a higher activity. This was observed in young and old. And here the activities of the complex 1 and complex 2 of the respiratory chain was observed increase just in young and not in old. So I don't know why, and authors just describe and suggest possibilities. So, and this is the final paper that I found that describe a metabolic shift from glycolysis in young mitochondrial respiration in old in fibroblasts isolated from young, 3 days of life, and old, 61 years old, fibroblast cells. And they measured the extracellular acidification rate as an indication of the glycolytic activity, and the oxygen consumption rate as indicative of the oxidative phosphorylation. This experiment is a classical experiment performed in the seahorse equipment. It's a very sensitive equipment. We can do this measurement by 20,000 cells. It's a small amount, but it's good for adherent cells, not the suspension like immune cells. It's possible to use, but it's more difficult. You can wash and the cells go away. But they found in these fibroblasts that increased extracellular acidification in young, suggesting a glycolytic metabolism, and here in oxygen consumption is the other way, with the older cells with a higher oxidative capacity, showing that, suggesting this metabolic shift with age. So, as a final remarks, it's my thoughts. I hope to have shown, I convinced you that mitochondria is more than the powerhouse of the cell, and they are sophisticated organelles playing high and complex varied process. And regarding mitochondrial alterations in aging, the question is how to move on, because it's necessary to move on in this field. I'd like to point the need for the establishment of a cause-consequence relationship, because I always say this: dying cells have dysfunctional mitochondria. But this dysfunctional mitochondria triggered the cell death process, or just the consequence of the cell death? It's important to try to distinguish between these both things. I think we have to improve the experimental models and models in age. I think it's not so easy, because this inclusion and exclusion criteria is difficult to control, mainly in humans, because you have to consider associated disease. And when people getting older, the probability of other associated disease increase. And in the review papers, aging is often considered along with neurodegenerative disease. And I don't know this answer. I think there is aging without ND, and there is aging with the neurodegenerative disease. And I think these things that have to consider in a separate way. But so I think it's is an open field to investigate the mitochondrial function in these models. So I would like to thank my collaborators, Professor Ivan from Unifesp, a collaborator from the past, Professor Valkyria, we start to try to do something. Professor Denise Arruda from UNC that helped me with the electron microscopy experiments. Professor Cesar that has the autobottle respirometer in UFABC. Professor Silvia Stucki from USP that give us the cell models. Professor Don Milmeyer, with whom we did these experiments related to MOMP, mitochondrial outer membrane permeabilization, and a new collaboration from the people from the Italy group, from Professor Salvatore Amoroso, Ancona, University Politecnica delle Marche, working with the sodium-calcium exchangers, controlling the ionic and metabolic, exerting ionic and metabolic control. We have preliminary data about this. And also the students that do the hard work, and also the funding agency. Thank you for your attention. I'm sorry about the time.
Very interesting. Thank you for the outstanding presentation.
Thank you.
And also for introducing the seahorse. That is the one that I'm using the most. And a comment on seahorse is that there are B-cell papers on aging mice and humans that you can add to your list of published data on humans. But this is B-cells, so probably
Yes.
you were more focusing on T-cells. And the other thing on seahorse is that the reason to, the only way to do seahorse on cells in suspension is to use the cell tack, because you have to attach, it's a kind of glue for people that don't know that you need to use in order to have the cells attached to the bottom of the microcartridges that we use in the machine. But I have a general question on the glycolytic phenotype of tumor versus tumor infiltrating lymphocytes, any kind of cell. For example, in breast cancer biopsies, I have a lot of B cells infiltrating. B cells make a lot of lactate, as well as the tumor cells. So there is a competition from the nutrients in the tumor microenvironment. So maybe the tumors get the most, the tumor cells get the most, and then the other cells, depending on what is the microenvironment, can be a good job as anti-tumor or pro-tumor effect. What is your thought about this? Which is the most important metabolite in the tumor microenvironment that is regulating this glycolytic effector function of tumor cells versus immune cells infiltrating?
Well, thank you for the question. It's a very complex one, and I don't know this answer, but these metabolites secreted by these cells in the tumor microenvironment are thought to be signals to one or another cells. And when we think about the tumor microenvironment, we have different layers of oxygen availability and substrates availability. And so I don't know about this. It's a very interesting question, because it's difficult to reproduce the tumor microenvironment. But more than the metabolites, I would like to add in your question the extra vesicles, extracellular vesicles.
Which is even more difficult to investigate, right?
Yes, and then I read a paper recently that cells can do mitochondrial transfer. One mitochondria to another cell, so it's so crazy. So I don't know the answer, but I think there is a lot of signals communicating these cells, and there are a very different situations in tumor microenvironment. I recently saw a lecture from Professor Elder from Einstein, working with single cell proteomic or cytometry, transcriptomic, single cell, try to isolate these cells from the tumor microenvironment. I think that there is a published paper, Professor Elder Nakaya.
Well, he was a collaborator.
Okay. Yeah.
Now he's back here, right? He's in São Paulo again.
Yes, Einstein Hospital.
I know him, yeah. More questions?
Yeah. When we were showing the muscle experiment, not experiment, biopsy actually, in my mind, it made a lot of sense, because when you're getting older, you're not getting only less muscle cells, but the quality of them also is very low compared to the young guy. So it makes sense to me if I believe that mitochondria is working on these cells or not working, then you have less strength when you are older. So you cannot walk a lot, or you cannot take heavy objects, because your muscle has less cells and not with good quality. So my question is, do you believe that this low quality of the muscle cells is related with mitochondrial function or numbers or fission or fusion or whatever? Just
Yes, it's a good point. It's my point here. It's a chicken egg tale here, because I don't know. People have to investigate this, because I don't know if mitochondria became dysfunctional and then leads to organization of the tissue, the organization of the muscle, or is the other way? So I don't know. We have to establish models to separate this, and the better situation is to see this kinetically, not the beginning and the end. So it's so difficult to establish these models and then do the experiments.
because from Maisa's data, you see that the people who has never had any complication, if they get the complication after 80s, they die quickly. So it seems to me that you have already mitochondrial damage or change or inefficacy. And then if you get sick, that's it. You don't have a mitochondrial recovery, or I don't know really mitochondria. I'm just guessing a lot of things here.
Yes. But what is the trigger? Mitochondria after 80s just start to generate more ROS. Why? Proteins are being oxidized along the time, and then after 80s, it's bad. So I don't know. It's a very interesting question. We have to.
but we have to think that it's intriguing that there are some elderly people that become to do exercise after 80 or 90 years old and become an athlete. And they can do a lot of exercise. They go to competition and they have good scores. And so if the mitochondria was not good, could not be possible to do that, no? I don't know. I think it's it's a big challenge to understand this. That's true.
So something about energy
Change. Something changed.
It's not just exercise. And your heart. What about your heart? And so many other neural dementia and so on. Is all based on mitochondria, or we don't know? That's so interesting for me, at least.
Yeah, but dying cells have dysfunctional mitochondria. The cause of or consequence is had to be assessed. And more than this, we have to consider in senescent cells the loss of the accuracy of transcription, because a lot of mitochondrial proteins are translated from the nuclear genes, not mitochondrial DNA. So this genetic the loss of this quality control of the replication, translation, so can generate bad mitochondria, energetically bad mitochondria. I don't know. It's not an intrinsic process in the mitochondria, or protein translated are not really good.
We need to remember that that mitochondria is very important to is a producer of substrate cofactors for epigenetic mechanisms, and epigenetic mechanisms are also involved in mitochondria function and environment change epigenetic marks, and I think this is an important point.
I saw recently a paper published in the JBC showing that alpha-ketoglutarate that came from the glutamate acting, controlling the histone acetylation, the acetylation, I don't know, but acting on epigenetics. Yeah. Thank you, Miriam.
Yeah, how you mentioned this.
Weak muscles, heart beating, heart attack, whatever. That's why people are so much to all this situation.
Yeah, but these are very complex process. So we have to, I think we have to do small questions and answer these small questions one step, because I felt tired to thinking about of this, and I can do the experiments to respond all of this.
Just to add the information that you get in the beginning of your talk about the caloric restriction. It was an in vitro study with yeast, yes. And in humans, we have the experience in one blue zone in Okinawa, Japan, where there are a lot of 80 plus individuals, and caloric restriction is part of the culture. And then it makes sense with epigenetics too, because it is an habit, it's a good habit, and they live with this caloric restriction for many years. And probably there is an association with the healthy life that they have. So what do you think about caloric restriction, epigenetics, and mitochondria? Is this a way to treat this dysfunction?
Yes, epigenetics, I don't know much about this. Professor Miriam will tell us more about this, but caloric restrictions induces the expression of uncoupling protein, the UCP in mitochondria. And these proteins dissipate the electrochemical gradient as heat and not ATP. But why? This uncoupling is also to generate heat, but also to decrease the ROS generation.
Yeah, I have a question.
Of
What is
course.
uncouple?
Sorry?
I don't know what is uncouple protein means. Oh, okay.
Yes, this protein dissociated the electron transport from the ATP synthesis. Okay. And this protein does this by dissipating the electrochemical gradient. Mm-hmm. And we can use substances like CCCP, dinitrophenol, but we have an intrinsic protein named the UCP uncoupling protein, highly expressed in brown adipose tissue. But plants also have this kind of proteins it's named pump uncoupling mitochondrial protein to fruits become mature, the green to tomato, green to red tomato, the expression of this protein is decreasing. So I think caloric restriction has this first impact in the expression of UCP. It's well established, but I think that there is other impacts. I don't know. Thank you.
Look at the guys that already taking the camera
Alô, som. Ei, som, teste, som.
Então, boa tarde. Retornamos com a... Good evening. Good afternoon. Let's return with our mini-symposium. And now I have the pleasure to introduce Professora Miriam Jasuliones. She's working on epigenetics, and she was very kind to come here today and give us a talk about her major interests, that is in cancer, but she's going to talk to us also about aging. Thank you, Miriam, very much.
Thank you, Valkyria. Thank you for inviting me to be here. It's a pleasure. It's a pleasure to be here with you in this wonderful mini-symposium. Well, as Dr. Valkyria said, aging is not has not been my research field, but I have studied cancer epigenetics, which is in some way related to aging. So I will discuss with you something about epigenetics and aging. Several different hallmarks have been attributed to aging. These hallmarks include primary hallmarks. These hallmarks have been related with damages affecting the genome. There are some hallmarks considered antagonistic hallmarks because they are related with responses to damage, and other hallmarks considered integrative hallmarks are a result of accumulated damage. Today, the focus will be on epigenetic alterations, which is a primary hallmark. Epi means beyond genetics, which means that there is an information on the chromatin, beyond that on the DNA sequence that is transmitted along cell division. Among the most studied epigenetic mechanisms are DNA methylation and post-translational histone modifications. These mechanisms add chemical groups to the chromatin. DNA methylation refers to the addition of a methyl group on the DNA sequence. And post-translational histone modifications refer to the addition of different chemical groups to the histone proteins in which the DNA is wrapped around. Both mechanisms alter the chromatin structure and play important roles in the regulation of gene expression and maintenance of genomic stability. In mammals, the DNA methylation occurs in CpG dinucleotides in the cytosine in the context of CpG dinucleotides. All cytosine in this context are considered potential target for methylation. The addition of methyl groups in the carbon 5 of cytosines is catalyzed by enzymes named DNA methyltransferases, or DNMTs. And this methylated cytosine can be recognized by proteins of the family of methyl binding proteins. The DNMTs 3A and 3B are considered de novo DNMTs because these enzymes are able to initiate methylation patterns. These enzymes can recognize any DNA sequence and add methyl groups. They play important roles during embryonic development because in this stage, epigenetic DNA methylation patterns are established. And DNMT1 is considered the maintenance DNMT because this enzyme has a preference for hemimethylated DNA, in which one DNA strand is methylated and the other is not methylated. This condition occurs during DNA replication. So during DNA replication, the original DNA strand presents some cytosines methylated, and the newly formed DNA strand is non-methylated. So this enzyme recognizes this hemimethylated DNA and copy the methylation pattern to the newly formed DNA strand. In this way, this enzyme plays an important role in the maintenance of DNA methylation during replication. So the DNA methylation pattern is replicated during mitosis. So the most known effect of DNA methylation is in gene promoters. More than 60% of all gene promoters present a high number of CpGs, known as CpG islands. In normal cells, most gene promoters are non-methylated, leading these genes permissive to gene transcription. However, when these promoters get hypermethylated, the transcription is inhibited. So the DNA methylation has a role in the transcription of regulation. So in normal cells, our genome has regions that must be methylated and regions that must be non-methylated. In general, our genome is rich in repetitive DNA sequences, retrovirus sequences, and these regions are in normal cells highly methylated. This methylation inhibits the expression of undesirable elements such as transposons, and also inhibits the homologous recombination between repetitive sequences. So the methylation of these sequences has a role in maintaining the genome stability. So we have in our genome other sequences that in normal cells are non-methylated. These regions are gene promoters. Most gene promoters are non-methylated. As I said, leading the genes permissive to gene transcription. Okay. So these are the two main functions of genome methylation: maintenance of genome stability and transcriptional regulation. Regarding histone modifications, as I said, these proteins, histones, in which the DNA is wrapped around, these proteins can be modified by different chemical groups, mainly in their N-terminal regions. There are several amino acids in these regions that can be modified by chemical groups. These histones can be acetylated, methylated, phosphorylated, ubiquitinated, sumoylated, and so on. And most of these modifications can be recognized by proteins that are, for example, transcriptional activators or transcriptional repressors, or proteins involved in DNA repair, or proteins involved in DNA replication. These histones are organized in an octamer. So the nucleosome is the structure that we know in which the DNA is wrapped around this octamer of histones. This octamer is constituted by two subunits of H2B histone, two of H2A, two of H3, and two of H4 histones. Each one of the subunits can be modified by chemical groups in different amino acids, especially in the N-terminal region. So some of these modifications are favorable for gene transcription, and other modifications are unfavorable for gene transcription. So in the nucleosome, the DNA region associated with this histone core is affected by the set of modifications in the histone core. Okay, so here there are some examples of active histone marks and some examples of repressive histone marks. This means that the histone, the subunit histone 3 is acetylated. Here, the amino acid lysine in the position 4 is 3-methylated in the histone 3, and so on. The histone modification machinery is composed by a huge number of enzymes and different proteins. There are enzymes responsible for adding the chemical groups in the histones. These enzymes are known as writers. There are another group of enzymes responsible for removing chemical groups from the histones. These enzymes are known as erasers. And there are another group of proteins known as readers responsible for the recognition of histone modifications. As I said, these proteins can be co-activators, repressors, or proteins involved, for example, in DNA repair. So these modifications, these histone modifications, have a role not only in the transcriptional activation and repression, but also in DNA replication and DNA damage repair. So as I showed for DNA methylation, our genome has regions that must present active histone modifications. For example, regions rich in genes, in active genes, these regions present active histone modifications. And regions, repressed chromatin regions, such as pericentromeric and subtelomeric regions, present mainly repressive histone modifications. So besides the role of epigenetic modifications in the promoter regions, here we have a representation showing a promoter region, unmethylated, represented by this blank, by these white circles. Here the transcriptional start site. Today we know that epigenetic modifications present a role when occurs in different sites of the genome, not only in the promoters. In the gene body, for example, DNA methylation in the gene body can favor gene transcription, can regulate the splicing process. We now know that for example, the methylation of sites where enhancers and insulators bind affect the binding of these proteins. There are a beautiful article that I like. I think this is a study published about 10 years ago. In this study, the researchers showed that the interleukin-2 expression is regulated by epigenetic, by DNA methylation in an enhancer region. They showed that in the naive T cells, there are two CPGs there in naive cells are methylated, and in T lymphocyte, when the T lymphocytes are activated with stimulus, these two CPGs become unmethylated, permitting the binding of proteins here that initiate, that activate gene transcription, the transcription of interleukin-2. And interestingly, in this study, they showed that the T cell that is not active, but I don't know how to say in English, the T cell that is not activated, but that saw the stimulus, that saw the antigen, regain methylation in one of these CPGs. One CPG remain, the protein remain binded in the CPG sites, and the other regain methylation. And this is the memory that the T lymphocyte saw the antigen. It's a beautiful study. So now we know that even a few CPGs can play important roles. Okay. So in a simple view, we know that active chromatin rich in active genes present active epigenetic modifications, and repressed chromatin present epigenetic modifications that is unfavorable for gene transcription. And these states are dynamic and are regulated by environment and also by intrinsic alterations. We know that epigenetic marks define gene expression patterns that are inherited, that are potentially reversible, and are susceptible to environmental influence. We now know that diet, inflammation, drugs, stress, maternal behavior, smoke, hormones can affect epigenetic marks. So now we know that the epigenome permits that the genome talks with the environment. These epigenetic mechanisms play crucial roles in physiological processes such as embryonic development, differentiation, genomic imprinting, X chromosome inactivation, memory establishment. And we know that with aging, epigenetic modifications change. And we know that several diseases are related with abnormal epigenetic marks, such as psychiatric disorders, autoimmunity, obesity, atherosclerosis, cancer. All of them are related with epigenetic alterations. So during embryonic development, the epigenotype, which is considered the totality of epigenetic marks of an individual, the epigenotype is established. All the epigenetic marks are established during this moment and are under the influence of environmental factors and also under the influence of intrinsic factors. And the epigenotype will define the phenotype. And we know that this epigenotype should be maintained along our life. But the maintenance of these epigenetic marks is influenced also by environmental factors and intrinsic factors, and may affect our epigenotype and also our phenotype. So our phenotype is a result of both epigenetic genetic information and epigenetic information, which is under the influence of environment. So during the embryonic development and lactation, we know that the mother phenotype, such as the size of the uterus, placenta, and also the environmental conditions, such as maternal diet, stress, mothering, can lead to epigenetic alterations in a way that a suitable environment is related to a regular development, and a damaging environment is related to metabolic and mental diseases in adulthood. In the same way, in the adult life, we are under the influence of several factors, several environmental conditions, for example, diet, stress, pharmacological treatments, unhealthy habits, toxic workplace, living place, in a such way that in a suitable environment, the aging can be delayed and the development is normal. But in a damaging environment, we have advanced aging and disease. So different conditions, as I said, such as diet, habit, different conditions, exercise, all of these conditions can modify, can modulate the epigenome. And these alterations can contribute to the early onset of aging and age-related diseases. Studies of identical twins, there are several studies of epigenetics in identical twins. There are studies showing that identical twins present their epigenomes very similar in early life. Here we can see that repressed genomic regions such as telomeres, repetitive DNA sequences not reaching CPGs, all of the sequences are highly methylated, and gene promoters reaching CPGs are unmethylated, as expected. But in the old twins, this pattern changes. We can see that many gene promoters gain methylation, regions that must be methylated lost methylation. And it's interestingly that these differences are more evident in identical twins that lived far one from another, showing the influence of the environment in this epigenetic pattern. And the extension of these epigenetic alterations now we know that these alterations can predict the chronological age. And this set of epigenetic alterations is known as epigenetic clock, epigenetic age clock. Okay, among the age-related epigenetic alterations, we have reduced expression of DNA MTs, leading to decreased global DNA methylation. We have, for example, increased DNA damage, such as double-strand breaks, which interfere with the landscape of epigenetic modifications. We have many metabolic alterations, and as we mentioned earlier, the metabolism is very important to give the cofactors and the substrates for epigenetic reactions. Okay, so the alterations in the metabolism leads to epigenetic changes. So in a general view, we can say that during aging, we have a global reduction in the heterochromatin. We have regions in our genome that gain methylation and regions that lose methylation. Mainly, the regions that must be methylated in normal cells lose methylation with aging, and regions that must be non-methylated gain methylation with aging. Okay, and we have also different alterations in histone modifications also during aging. So these age-related epigenetic alterations can be detected both in different tissues and also in the blood, and can use it as markers. Okay, so the young epigenomics is favor a phenotypic plasticity, contribute to a phenotypic plasticity, and with a balanced responsiveness to environmental stimuli, contributing to a healthy life. However, during aging, it's observed an epigenetic drift that causes an increased noise in gene expression. And this is observed in aged epigenomics. So aged epigenomics is related with a decline in phenotypic plasticity and a decreased responsiveness to environmental stimuli, contributing to the emergence of diseases. So here we have a brief timeline of age-related genetic alterations. The first observation was done in 1967, where the whole genome DNA methylation was associated with age in spawning salmon. In 2010, a specific histone modification was linked to aging in metazoan. In 2013, it was proposed the first epigenetic clock to predict biological age. And later in 2017, some RNA modifications were associated with aging. And in 2018, single-cell chromatin modification profiling elucidated mechanisms of epigenetics and aging. So since the first proposal of an epigenetic age clock to predict the biological age, several other epigenetic clocks were proposed. All of them are based in a number of CPGs in which the methylation status is able to predict chronological age and also the onset of age-related diseases and mortality risk. These modifications can be detected both in tissues and in the blood. So among the risk factors able to accelerate the genetic clock are hereditary diseases, neurological infection diseases, and also adverse lifestyle such as stress, increased BMI, traumatic events, disturbed sleep patterns. And among the factors related to decelerating epigenetic clock are also some hereditary factors, enhanced synthesis of epigenetic enzymes and cofactors, inhibited mTOR, and beneficial lifestyles such as high income, education, regular exercise, fruit and vegetable consumption, and sufficient sleep. So epigenetic changes are crucial, contribute to the loss of heterochromatin, which is a characteristic of aging, the loss of heterochromatin. So maintaining the balance between heterochromatin and euchromatin can avoid the cellular defects that accumulate with aging, such as genomic instability, mitochondrial dysfunction, metabolic imbalance, chronic inflammation, and so on. There are rejuvenating interventions aiming to delay aging and avoid age-related diseases. These rejuvenating interventions work through different layers of epigenetic regulation in order to lead to a healthier state in cells, tissues, and organs. So here we have a brief timeline of aging delaying therapies. The first one was proposed in 1935, which is the caloric restriction mentioned today, earlier. We have interventions based on the activation of sirtuins, based on circadian rhythm in cellular reprogramming, based on the elimination of senescent cells, named senolytic therapies, and also aging vaccines. So one of these rejuvenating strategies is based on a metabolic manipulation, the caloric restriction. We know that metabolism and epigenetics are strongly related, strongly linked. And both metabolism and epigenetics influence the aging process. So the caloric restriction alter epigenetic marks, and these alterations result in alteration of genes, in the reset of a normal expression during aging. I cannot see. Alter, I think mitochondria also play a role in heterochromatin also. Another rejuvenating strategy, as I said, is the cellular reprogramming. Some studies show that fibroblasts collected from centenarians, supercentenarians, or patients with progeroid syndromes can be used to successfully generate induced pluripotent stem cells. And this cellular reprogramming depends, the efficiency of this cellular reprogramming depends on an epigenetic reprogramming. So a number of pharmacological and environmental interventions longevity interventions have their beneficial effects depending on epigenetic alterations. Here we can see that several of these interventions alter epigenetic enzymes or alter substrates or cofactors important for epigenetic modifications. And these interventions able to rejuvenate the epigenetic modifications also reverse other hallmarks of aging. For example, genomic instability by avoid epigenetic changes related to aging. It's possible to avoid genomic instability. It's possible to decrease chronic inflammation, avoid mitochondrial dysfunction. Mitochondrial dysfunction is frequently observed during aging. And we know that the environment can change epigenetic marks, and these epigenetic marks can change the expression of mitochondrial genes, affecting the function of the mitochondria. So in normal conditions, epigenetic marks affect contribute to the normal function of mitochondria. And the mitochondria, as I said before, is responsible to supply several cofactors and substrates for epigenetic mechanisms. So one depends on the other. And the rejuvenating strategies, several of these interventions are able to influence this communication, leading to a younger state of mitochondria. So, as I said, in young cells, the heterochromatin is present. The DNA regions that must be condensed are condensed, and this heterochromatin is lost with aging. This loss of heterochromatin contributes to genomic and epigenomic instability, to translocations and genetic alterations, and also with the aberrant expression of repetitive sequences. And the loss of heterochromatin is a result of epigenetic alterations. Oh, sorry. And here we know that several rejuvenating interventions can contribute to old cells to maintain a heterochromatin, to avoid the loss of heterochromatin. So the loss of heterochromatin leads to the expression of undesirable elements, as I said, for example, heterotransposons, which can go to different DNA regions, leading to DNA damage and with double-strand DNA. And both double-strand DNA and mitochondrial DNA, as a result of damaged mitochondria, both can activate cGAS-STING pathway, which induces the expression of interferon, senescent-associated secretory phenotype, and can culminate in the chronic inflammation. So by using the rejuvenating strategies, both the mitochondrial dysfunction can be inhibited, as well as the loss of heterochromatin, avoiding the instability and the chronic inflammation. So different rejuvenating strategies, for example, I showed metabolic intervention, the caloric restriction, cellular reprogramming, the use of life-extending chemicals, or the ablation of senescent cells, interfere with different processes. For example, the metabolic intervention induced the rejuvenation of stem cells, induce anti-inflammation, retro element repression, and mitochondrial rescue. Most of them act in different effects, and these effects involve epigenetic rejuvenation. Okay, so I hope I can convince you that epigenetic is involved in aging and in interventions aiming to delay aging. So to finish, I would like to let this message. This was published in a review from these authors, Peter Jones and Martisen, that said that epigenetic marks can be described as marks of experience. They contribute to differentiate us from our younger selves. Thank you.
Thank you very much, Miriam. Very nice presentation. I enjoyed it very much. So, any questions?
Yes, very interesting presentation. And actually, you convinced us that there is an epigenetic problem with aging. My question is on the epigenetic rejuvenation that you have proposed can be obtained at least experimentally, let's say, with in different ways. And one of these is for example, with caloric restriction or with, let's say, metabolic control. Another one is senolytics. You cited some D plus Q plus quercetin, which is the same mix that we use. But actually with these senolytics, the effect is transient. Mm-hmm. So when you, for example, treat people that are obese with type 2D with these compounds for 14 days, you analyze the beta-gal positive cells, the senescent cells in any tissue, for example, the adipose tissue, in biopsies. Before and after the 14 days of treatment, you have a significant decrease, a drop in the number of senescent cells. But if you interrupt the treatment, these cells, little by little, start to come back. Is the same kind of result expected with metabolic modifiers, metformin, or with caloric restriction? Is it known for how long these effects are lasting?
I don't know, because I'm not an aging researcher, and then I have to read more about this to respond this question. But making a parallel with cancer, in cancer, there are some genetic treatments based on inhibitors of genetic enzymes, for example. And we know that, for example, the methylating agents are able to demethylate the DNA, and after the removal of this treatment, some genomic regions regain methylation, but other regions no. Same can occur. I don't know because I need to read about this too.
So it's not on what is determining in some tissues or people the duration of the intervention.
I imagine that it's possible that the same occurs with these treatments. Maybe some epigenetic alterations remain and others not.
Thank you.
Thank you, Miriam, for your nice presentation. And here we see the background of all the good habits that we have to incentivize in our patients.
Yeah.
So even in this population of 80 plus, it's time to modify a lot of things. So I'm following these patients for 1 year now with Dr. Maisa, and we see that increasing physical activity, it can be walking or some resistant exercise, improving the diet, it makes a lot of difference. Of course, if the patient has dementia, maybe it will not happen. But even a patient with a lot of comorbidities, all of these small modifications can give some increase in quality of life. So I totally agree with you. I think that this is the major area where we can act to improve
prevention. Yeah, I believe that several conditions can be stimulated to avoid diseases. So as you said, we know that the exercise, diet, we know less stress, we know which is good for us, yeah, and we know that all of these activities is really are related.
And this is the parallel with cancer patients, because the marks that we have to increase or avoid are the same. So I think that oncology and aging are completely connected in these hallmarks.
Yeah, we know that aging and diseases have both genetic and epigenetic components. But at least for epigenetic components, we can revert a normal epigenetic marks. And I think that we need to do this. We need to try to revert the normal genetic modifications.
Thank you.
And I believe that DNA damage is a very, very important aspect in aging and diseases, in cancer. Because for example, sirtuins, caloric restriction activates sirtuins. There are some resveratrol and other components are able to activate sirtuins. And sirtuins are enzymes, are deacetylases that deacetylase both histones and other proteins. And in normal conditions, sirtuin is associated with repressed chromatin. And there are studies showing that under stress, sirtuin is removed from the sequences. 10% of sirtuin remain in these sequences, and other relocate to other sequences. Sequences silence in aging. And I think that this is very important. And sirtuins are proteins recruited to DNA damage sites, DNA double-strand sites. And there is a study where the researchers show that they are able to induce a double-strand break in an exogenous promoter of E-cadherin. And they induce this double-strand break in the promoter, and they show that enzymes responsible for the DNA repair are recruited. Sirtuin-1 is recruited, and sirtuin-1 brings together epigenetic enzymes. And they showed that although 99.1% of cells are repaired okay, the DNA sequence is repaired, okay, but 0.1% of cells, although the DNA is repaired, these sequences present abnormal epigenetic alterations. Then it's possible that under continuous stress, under continuous DNA damage, cells acquire abnormal epigenetic alterations. Depending on the DNA in the genome sequence, these alterations can culminate in disease because the DNA damage. The alteration is not genetic, it's epigenetic.
But it's more or less what Daniela told us about obesity. Once you imprint this on your system, it's more or less like even though you have a small number of cells that you can grow drastically if you are exposed to some stressful situation.
Yeah.
I was just curious about therapies to remove senescent cells. I tried to read about that, but I didn't have much time. But it seems to me that, okay, they are senescent and we remove them, maybe we improve something, but I think in our older organisms, they might add some function. Not just bad function, but some, because if you remove them, I think the organism has to have a certain number of cells, so you're going to have peripheral homeostasis. So this peripheral proliferation to get again these numbers again on the periphery. But what are the cells who is going to be replaced to substitute the senescent cells we removed? Naive ones with for new antigens and so on, or the memories or everything at the same time. So I don't know. It's just a point to discuss.
No, you go ahead. So it is really not known which is the target in a pool of immune cells, which is the primary target of these senolytic treatments. First of all, you need some senescent cell, some SASP, because you need to get rid of certain kind of cells. So the SASP is also a protective mechanism. When you have a transformation going on, you have the senescent cells that stop the proliferation, and these cells may be eliminated. Unfortunately, with aging is the other side of the balance that is prevailing. So you have more negative effects of the senescent cells. Now, in the clinical trials that are under the control of Mayo Clinic and Kirkland and Ticonia, they're the ones that have, patented all these senolytics. Actually, they are looking at the at how much the age at which people get cancer is postponed as compared to people that are not taking senolytics. This is one thing. How much inflammation you have, I have some data, I think, now to show you, just to introduce the senolytics. How much inflammation in serum you can have under control if you take senolytics. But again, the purpose now is first of all to find the good combination of senolytics that have the longer effect, because you cannot be on senolytics all your life. This is very clear. Kirkland came 1 month ago to UM and said, don't take the senolytics by the counter, by the pharmacy, have them referred by a doctor, and don't do a continuous treatment with senolytics because you risk to get rid of the cells that you need with a SASP phenotype to protect from development of tumors and things like that. So that's the problem. The future research on senolytics is to find better molecules that can have longer effect and no side effects, because long-term treatment with senolytics actually have some metabolic unbalance in all the components of the basic metabolic pathways that the organism needs in order to make energy. For example, the muscle needs a minimum amount of energy to function. Well, the senolytics are impacting this equilibrium. So search for the best mix and also for new small molecules that can be active.
At the same way, you need somehow some amount of stem cells, or you need to have a small number of T cells. We really need to have some changes, some epigenetic changes to be postponed, not dramatic changes, but few. Is that necessary for us? Because if there are these mechanisms, are they necessary for some reason?
I don't know, maybe, I don't know.
Maybe in cancer or any other thing.
In cancer? Yeah. Yeah.
Has nothing to do with prevention or nothing to do, it's just to prevent things.
No, epigenetic marks are fundamental to the normal function of cells. The problem is a normal epigenetic patterns.
Yeah.
A normal epigenetic changes that when the epigenetic marks are present in the wrong place, they can contribute to disease. Yeah.
Yeah, no idea. Yeah, because I don't have time to read about all those things, but how are they?
Well, Mary-Rose has a lot of data on the reprogramming of the epigenetic clock and making actually, he was the first one describing that the epigenetic clock was showing in centenarians a biological age that was different from the chronological age. So, which are the mechanisms that he has scored in centenarians versus the siblings that are just 80 and older? I don't know. I should know.
The lifestyle. Maybe.
Less harmful.
Yeah.
And we have to remember that everything is therapy. So you cannot do it forever. It's used for treatment of chronic myeloid leukemia. It's an inhibitor of tyrosine kinase. So you have to use for a short period of time. In this sense, maybe metformin that acts for another mechanism can be less harmful in the same way that you are using, for example, hypoglycemia drugs used for diabetes type 2 to treat obesity. Mm-hmm. So metformin maybe in the future can be used in health lifestyle aging. I don't know. Yes. Yes. Yeah. Here in Brazil. Yeah. Here in Brazil, it's not the reality, but we are coming to this point.
I just want to know about caloric restriction. You all mentioned it. So what are you talking about in reference to what? Can you quantify it? Like, what do you call caloric restriction?
Which condition?
You all mentioned caloric restriction. So
you mean how?
What is it? I mean, caloric.
I think at least 20% reduction.
20% in relation to what? To your daily, baseline, you ingest?
Yeah.
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 In humans, they have accepted a little bit less because it can be too much. Maintained over a period of 12 months. Mm-hmm. So there is the calorie study from Yale where actually they are reducing the intake of nutrients by 5 to 10% in 6 months. If this is maintained over the second 6 months of the year, caloric restriction was lower than the 20 that was initially described. It's too much.
Yeah.
Birthday on 365 days.
Muscles, muscular tissue. So if the diet or the restriction is too high, so yeah, it's not indicate for everybody. So. Mm-hmm. Yes. Mm-hmm.
For the population in general, or just in study? Well, no, that was a study including thousands of patients between Connecticut and Oklahoma. The 2000, I don't know if you remember, dietary restriction, the responsible of the study was big city. I think there were a little bit more than 1,000. That was just published in Nature. Low impact factor. Like this last year. That's a lot.
Yeah. We fight against the excessive calories. Yeah.
Modulates the epigenetics. Is it an allosteric modulation of enzymes related to methylation or acetylation?
For example, how diet interfere with genetic marks. The universal donor of methyl groups are S-adenosylmethionine, from the cycle of methionine. Methionine is converted to S-adenosylmethionine, which donor methyl groups for all methylation reactions to methylate histones, methylate DNA, proteins, and so on. After the donation of methyl group, the S-adenosylmethionine is converted in S-adenosylhomocysteine that can be transformed again in methionine. This methionine cycle is coupled with folate cycle, and this is also linked with the transsulfuration pathway to produce glutathione. All of these reactions depend on cofactors, for example, B6 vitamin, B12 vitamin, amino acids, methionine, and so on. The diet interferes with methylation, for example, in this way. The acetyl group comes from acetyl-CoA, from the metabolism. So all of the metabolites act as substrates or as cofactors in these reactions and affect the donation of methyl group and acetyl, for example.
I think it's easy to imagine in the case of the cofactors, but in the case of alpha-ketoglutarate, it's intriguing because
alpha-ketoglutarate. I don't know if I know how to explain this, but there is an alteration in TCA cycle. I don't remember the substrate that become alpha-ketoglutarate. Don't remember, but there are an alteration, and this alter the activity of several epigenetic enzymes. That depends on.
It's an allosteric modulation. I don't know, because I saw a paper with altered alpha-ketoglutarate and the pattern of these enzymes responsible epigenetics. But I was wondering how
I know that
mechanisms
they are they act as cofactors, but I don't know how they interfere. I don't know. I need to read. I don't know.
Just curious. Thank you.
Thank you. Okay.
Have you studied any old mice or old cells?
No.
No.
We can do this.
Okay. Thank you very much.
Thank you.
For 8% are over 2 years. We have more than 2,000. The paper is in Science. Spada, my last name is Dixit, T-I-X-I-E-T.
So now, just to finish this enlightening symposium, I'd like to call again Daniela Frasca to give us one more talk about her results on aging. Thank you, Daniela.
And very interesting day here, very interesting discussion, and a lot of new ideas. Thank you all for this. This is there. Okay, let me see if I can do something different. Okay, I'm gonna show you now some ideas about aging, immunosenescence, and how this phenotype, the SASP phenotype in immune cells, is regulated immunometabolically, which is another interesting aspect of my studies. So immunometabolism is something that now is very timely. Everything is regulated in the immune cells, metabolically speaking. These two things were completely separated until 10, 15 years ago, with the metabolism regulating the nutrient uptake in the individual and immune function regulating the response to infections and vaccines. But now the two things are very integrated. And it has been shown in the last years that the effector function of immune cells is linked to cell metabolism, which is strictly dependent on the metabolism of the individual. It has also been shown that the activation of different immune cell types is supported by a metabolic reprogramming. And this can be a good or a bad thing, because sometimes the metabolic reprogramming is supporting some processes that are not good for the organism. And of course, from these initial evidence findings, it come out the possibility that you can actually modify some metabolic pathways in order to improve immune function, for example, protective responses to infections and vaccines, and block pathogenic functions. And of course, as a consequence, metabolic enzymes, some of them have already been described to directly regulate a lot of immune functions. So aging is associated with changes in the metabolic profile. In the serum, the phenotype of an individual, metabolically speaking, is different if you are 65 years and older from a younger age. from your younger controls. But unfortunately, there are no studies published in humans in healthy aging comparing the metabolic profile of serum samples from young and elderly individuals, or even middle age versus young or old and centenarians. Everything is associated with disease. Everything is associated with gender bias studies. So actually the results, the better results, are still the ones obtained in mice many years ago, but confirmed throughout years in different strains. And these are completely healthy mice, young and old mice, where this is just a simple two-way hierarchical clustering of total metabolites that are upregulated or downregulated in the serum, in serum samples of mice of different ages. I'm sorry, but I cannot see. So what came immediately evident was that there were several pathways, metabolic pathways, that were activated in old versus young mice. And these pathways indicate that the lactate dehydrogenase, which is an enzyme important for the generation of lactate, as well as the two types of cholesterol and the total cholesterol, glucose, the amylase, or the serum amyloid protein, as well as fatty acids, free fatty acids, were upregulated in the serum of old, healthy mice as compared to young controls. And at the same time, there were some pathways like triglycerides and creatinine that were downregulated. So when these people did a more specific and targeted metabolomic analysis, so they just did the same hierarchical clustering, but only of targeted metabolites, they found, as you can see here, a lot of long-chain saturated fatty acids that were present in old but not in young. And that was confirming once again that this is one of the major changes in almost all the animals that were evaluated in different strains, in females versus females. So all these studies confirmed that the fatty acids are actually among the most abundant metabolites in the serum of aging mice as compared to young controls. So some of these came out from studies on inflammaging as metabolites that may regulate this systemic chronic low-grade inflammatory status of the elderly. And these in particular are, so first of all, we did, you see today we have the arrows. We did the analysis of these markers that we have seen in mouse studies, and we did a principal component analysis, which is just a statistical way to separate clusters of individuals. And you can see that based on this, LDL, which was the one showing the less difference between young and old mice. And all the others, HDL, glucose, fatty acids, lactate dehydrogenase, CRP, which is the marker of inflammaging, and the serum amyloid A. So based on this analysis, the elderly and the young in this biplot were completely separated. Of course, again, some markers had a different weight from some others, like LDL. But many of these, like lactate dehydrogenase and CRP and serum amyloid A, are markers of inflammation and inflammaging, so were initially described as markers of inflammaging. So we tested them in our cohort of 20 young and elderly individuals, and here they are. They are higher. The three of them, these are the ones that we focused more, because it's something that we now do routinely to test these markers, to have an inflammatory signature of the serum of the participants in our study. And when we studied the response to infections, our cohort of younger and older individuals infected with coronavirus of the recent pandemic, you see that the lower antibody responses were associated very significantly with higher levels of LDH, lactate dehydrogenase in serum. And similarly, the influenza vaccine response, when there is a lower response, this is in many cases associated significantly with there's a negative association with plasma levels of lactate dehydrogenase. So actually in many other studies, including the Callery study that I was talking about, the lactate dehydrogenase is one of the markers that is mainly considered when we talk about metabolic changes occurring with aging, diet, and conditions and diseases. Okay, so I don't have yet the results of our study, the metabolic analysis of serum samples from 20 young and 20 elderly, completely healthy individuals. It takes 6 months to have these results. It's a mass spectrometry analysis done at our facility. But, you know, the experiment is run very fast. I mean, it's 24, 48 hours to run the sample, and then 6 months to have the biostatistical analysis done. So, and this is extremely, extremely expensive, but this is with a person that is included among the collaborators. So he's the director of the Bascom-Palmer mass spec facility. So as a collaborator, he made us spending only a few $1,000s per sample, which is going to be anyway a lot. But why is this important? Because like inflammation that in serum is shown by several markers, like inflammation talks and induces an inflammatory profile in the immune cells, also the metabolic analysis of serum indicates markers of metabolic markers that may affect the metabolic phenotype of the immune cells, which is strictly connected to the capacity of the cells to respond in a good or bad way, doesn't matter, but to be effector cells of an immune response. So we analyzed by seahorse the metabolic phenotype of immune cells, B, T, and monocytes. We did CD4 because these are the cells that participate in vaccine responses. So we did also CD3 and CD8. But we used the seahorse technology that was this morning already introduced. This is the seahorse technology is used to evaluate in real time changes in oxidative oxygen consumption rates, which are a measure of oxidative phosphorylation, so mitochondria analysis, as well as extracellular acidification rates. So the machine is measuring this in real time. At the same time, you have these two types of graphs. This is mitochondria. This is cytoplasmic generation of lactate that is inducing a change in the pH in the cytoplasm, depending on the amount of lactate that is made. So these are sorted, B and CD4, and then loaded in seahorse. There is no activation here. The only difference between the two is the age, with the elderly on top of the young. The principle of this mitostress test, it is a mitostress. We load the cells and we inject in the micropores of the cartridge different components. First of all, we inject oligomycin. Let me explain you. We put the cells, we attach the cells to the bottom of the cartridge using this glue, cell tack, because we cannot measure these things in cells that are suspended. They have to be spun down. And then on top, there is another part of the cartridge where we can add the different chemicals, any kind of chemical that we want to use. But in a mitostress, we use these three components. Oligomycin. Oligomycin is blocking ATP synthase. So is from basal respiration, the cells have their own basal respiration. Blocking ATP synthase, you have a crash in ATP production. Okay. After a number of minutes, the machine gets the second compound, which is FCCP, is an uncoupling agent, which allows maximum respiration to occur. So this is a measure of maximum respiration. that is then blocked after a few minutes by these two compounds, antimycin A and rotenone. Here shown the effects of all these components directly added to cell culture of yeast cells or other cell types. So at this point, the respiration, the maximal respiration, comes back to basal levels. And what is very important is not only the maximal respiration or just the profile of the cells as it is shown here. It's also important to calculate many other values that the machine, the software, is generating automatically. So once you get these graphs, you also get how much basal respiration you have in one cell type versus the other, how much ATP is produced, although it's not, I learned today that this is not the best way to measure ATP. It's much better to have it in cells in culture. You have maximal respiration, spare respiration, which is the difference between maximal and basal respiration for each cell type. Okay, so we have this information in real time, mitochondrial function, and lactate production in the cytoplasm. So you can immediately see that there is a difference in both measures in B cells between young and elderly. So the B cells from elderly, healthy individuals are higher. I call them hypermetabolic. They have an hypermetabolic phenotype. The same happens with the CD40 cells, but you see the difference between the same y-axis. So these cells are less metabolic compared to the B cells, unless when they are unstimulated. And I'm interested in the non-stimulated phenotype because higher is the level of the metabolic and associated senescent expression, marker of senescence expression in these cells before they are stimulated, lower is their capacity to respond well, or higher is their capacity to be pathogenic. So there is a strict correlation to the phenotype of the basal cells and their capacity to respond later. So B cells are highly metabolic, higher than CD4, and hypermetabolic in the elderly as compared to the young. Monocytes don't care. Look at the y-axis. These are super metabolic independently of anything else. That's why you have to get rid of monocytes when you do these studies, because if not, they take everything. Like tumor cells, they eat all the possible nutrient that is available. So, as you know, my interest is on B cells, and especially on those subsets of B cells, again, the double negative, that are the ones that are expanded in inflammatory conditions and are the more pro-inflammatory. So more pro-inflammatory means that they have the capacity, they must have the capacity to secrete pro-inflammatory mediators, for example, inflammatory cytokines or inflammatory metabolites, because they have to and they have to be, of course, supported by a metabolic reprogramming, because if not, in a very inflammatory environment, which is aging, adipose tissue, I don't know, the heart under obesity conditions, tissues that, you know, are in an inflammatory environment, these cells should die. They are terminally differentiated. They're, oh, no, don't go, no. They are terminally differentiated, but still transcriptionally, because they make cytokines. You know, I showed you this morning that these cells in obesity make antibodies, make pathogenic autoimmune antibodies. So they need to switch to a positive metabolic phenotype in order to survive. If not, the apoptosis is killing them. There is a lot of cell death, as I showed you this morning, also in aging. A lot of fatty acids around. Okay, so the double negative are the cells that express the higher levels of transcripts for the SASP, for the senescence-associated secretory phenotype. So they express the highest level. These are sorted from the peripheral blood of young and elderly individuals, healthy, again, no stimulation. They are sorted, and then by fluidyme, which is a qPCR way to measure expression of transcripts in as little number of cells as 100. It's not a single cell RNA-seq which is very expensive. This is less expensive, but at least we have not bulk big cultures. So you see that the expression of transcripts for TNF-alpha and for IL-6 and IL-8, these are very inflammatory markers associated with the SASP, are expressed only in memory, but especially in double negative B cells. The same pro-inflammatory microRNAs like miR-16 and miR-155, extremely highly inflammatory. And of course, the canonical marker of senescence, which is P16. So what do they do in order to support this function? Well, we found in 2015-16, in a collaboration with Arne Akbar in UCL in London, he was doing the T cell part, the CD4 T cell part, and we were doing the B cell part. We found the activation of this crucial protein, AMP activated protein kinase, which is a protein that is spontaneously activated in double negative B cells from the elderly as compared to the young, but especially is activated only in double negative and not in the other B cell subsets, suggesting that there is a preferential program that keeps these cells alive and makes them able to signal in order to make whatever they have to make, inflammatory products as well as autoimmune antibodies. And the interesting thing is that AMPK is regulated by metabolic stimuli. So unbalancing the ratio AMP-ATP, hypoxia as well as changes in intracellular calcium, inflammatory signals like TNF-alpha and IL-1, oxidative stress. So all these signals that are typical of all the cells convert on AMPK, which is then phosphorylated. It downstream phosphorylates this P38 MAP kinase in these particular residues. And in these conditions, there is no class switch, so no protection, no secretion of protective antibodies, and a lot of intrinsic inflammation in the double negative. So that was exactly the same kind of result that Arne got on CD4. CD4 T cells have a double negative, it's not called double negative, but has a double negative population that is not expressing CD27 and CD28, or CD57 and 28, I don't remember. So it's double negative for some markers of the T cells, and they activate this pathway of signaling. That was the first time that senescence and metabolism were shown to work together in immune cells. He published in Nature Immunology, and we did in experimental gerontology, because when we submitted to Nature Immunology, the reviewer said, oh, forget it, the B cells are not regulated metabolically. So he published there and we didn't. But okay, you need to be lucky in life. Okay, these are the results of AMPK. So what we did here, we sorted this is only elderly, because from the young, we don't have enough double negative to do this, to do at least Western blot. So we sorted double negative, and we sorted B cells without double negative that were used here. So this group here contains naive, IgM memory, and switch memory. So it's very clear that only double negative have phosphorylated AMPK and phosphorylated P38 MAP kinase, as well as other molecules that are also regulating, that was shown years later, molecules like the PI3 kinase, especially the P85 subunit, that is regulating the balance in the cell between anaerobic glycolysis and oxidative phosphorylation. But at that time, we didn't know that those results were so interesting. Okay, so spontaneous activation of these energy sensing pathways in B cell subsets that make the highest level of senescent markers. So then we tested, we compared, still in seahorse, we compared the metabolic phenotype of double negative versus the other cell types in the B cell pool. still from elderly, because again, we don't have enough to run these experiments. And again, we did oligomycin, FCCP, and rotenone and antimycin A treatment. And the bottom line is that, again, we have in the mitostress test a higher metabolic phenotype of the double negative versus the other cell types. And especially the glycolysis, the anaerobic glycolysis pathways go up. So these cells, like many other effector cell types, upregulate anaerobic glycolysis. And this is a problem, of course, when these cells infiltrate tumors, infiltrate the adipose tissue. Because if these cells under inflammatory conditions expand and they have to do their job there, these cells secrete lactate. So they contribute to the acidification of the medium, which is, of course, not a well thing in order to respond to cancer therapy, but also to get rid of the cancer. Okay, so double negative, at this point, after showing that the double negative are the highest in the metabolic phenotype compared to the other subset, we started analyzing and comparing double negative from young and elderly individuals. So we measure, first of all, this pathway. You know, because of the increase in ECAR, which is the anaerobic glycolysis, we started analyzing the pathways of anaerobic glycolysis, just looking for simplicity, even because we are not a biochemistry lab, very experienced like his lab. But we measure the glucose uptake. In this case, by measuring by qPCR the expression of the transcripts of the major glucose transporter, which in B cells, in humans, is GLUT1. And then we measure, of course, the lactate dehydrogenase, which is the enzyme that is activated in ECAR. So ECAR is anaerobic glycolysis, and this is the transcript that better represents the results of ECAR. And this is the enzyme that leads to the secretion of lactate. Okay, the results show that there is, of course, the highest level of GLUT1 and lactate dehydrogenase A are in double negative as compared to the other cell type from the elderly. There is also a significant difference with the expression in the young. We also look at this marker. This is a marker of oxidative phosphorylation. The primer is for one of the, I think, 3 or 4 complexes in the pyruvate dehydrogenase complex. Is made by 3 or 4 proteins, I don't remember now. And this is involved in the secretion of ROS. And even here, we have an increased expression of this transcript in double negative versus the other cell type. But you remember, OCR was higher in the double negative as compared to the other cells. So it was expected that we would find also a high expression of this marker, not as much as the lactate dehydrogenase. So now we started looking at the double negative. We sorted double negative from young and elderly individuals. Unfortunately, again, very few from the young because we don't have enough cells, and we did mainly flow cytometry analysis. So we analyzed the glucose uptake using this fluorescent analog 2-NBDG. And here we show I show the MFI of glucose uptake in the elderly is higher than in the young. And this is the graph showing all the evaluation by MFI. This is the other measure, the qPCR evaluation of GLUT1 expression, higher, and of course the two are associated. This is lactate dehydrogenase, so upregulated by qPCR. Look how much lactate is made by this double negative. So they're really secreting autoimmune pathogenic antibodies, pro-inflammatory cytokines, and now also pro-inflammatory metabolites. I will show you later that this is a pro-inflammatory metabolite. And again, lactate dehydrogenase A by qPCR is associated, as expected, with the secretion of lactate. And then we do, again, by fluidyme, a bunch of enzymes that are in this pathway that leads to the activation of lactate dehydrogenase and lactate secretion. And you see that the double negative from elderly have much more red color than the one from young. This is the glucose transporter. Exokinase 2 is another one that we have evaluated that is very interesting for some cell function. I don't have time to talk to you today. The ALDO enzymes, the NO enzymes, and of course LDHA. All of them are upregulated in the double negative from old as compared to young. And of course, because of the OCR data, we also wanted to check the amount of ROS that was available in this double negative from young, red, from old in red versus the young. You see there is, by flow cytometry using the Serrox, we were able to find more Serrox in the double negative of elderly, as well as higher levels of the enzyme PDHX that were, of course, correlated as expected. Now, this morning I introduced you to the concept that B cells are not only inflammatory, but they can also polarize other cell types towards a pro-inflammatory phenotype. So we did the following series of experiments. I keep here this cartoon just to show you what the pathways are. So from glucose, I'm showing here glucose, can be any other nutrient, of course, but you know, from glucose there is the glucose transporter can be measured. Then skipping all the pathways in the cytoplasm, we arrive to pyruvate that can be converted in lactate with a lactate dehydrogenase A, or can go into the mitochondria and generate ROS and regulate other pathways in the mitochondria. So the experiment is the following. We took CD4, so we wanted to see if CD4 T cells were supported and were induced to be inflammatory in the presence of B cells from young and elderly individuals. So we took CD4, we sorted CD4 from young individuals, and we put them in the transwell together with B cells from young and elderly individuals. And after 24 hours, we isolated the CD4, we isolated the RNA from the CD4, and we measure some metabolic markers, the major ones. We measure many others, but we measure the glucose transporter, lactate, and pyruvate dehydrogenase. We know that these are the pathways affected based on the seahorse results. So when we have this condition, actually there is no change in CD4 after they have been exposed 24 hours to B cells from young individuals. But if they are exposed to B cells from old individuals, you see GLUT1 goes up, lactate goes up, and not so much pyruvate. So still is the less activated pathway. It is activated, there is a significant effect as compared to this, but not as much as the other two. So this suggests that there is some secretion of lactate in these cultures. And in fact, this is the secretion of lactate in culture. This is without any stimulation. So the CD4, in the presence of B cells from old people, are stimulated to secrete lactate. Similar to what happens when you take them and you put them in vitro with a mitogen, a T-cell mitogen. They make lactate. And this is something that actually I was not expecting. I was expecting the lactate coming only from B cells. No, actually they also make it in this condition. And they make this, and of course, because they make lactate, they become inflammatory. So they secrete, this is picograms per ml of IL-17 and interferon gamma. And of course, much more in this condition than in this. There is some effect here. You see some of them have a twofold increase before and after, but much more in this condition. So we tried to block this pathway. that seems to be mediated by lactate for sure, and who knows by which other metabolites. So, and we use this molecule. It's a small molecule, FX11. This is a full name, forget to remember the name, but this is an inhibitor of the enzyme. So in the presence of FX11, there is no production of lactate because the enzyme is inhibited. The activity of the enzyme is inhibited. And in fact, in the presence, if these cells are pre-treated with FX11 for 24 hours, then the inhibitor is removed. There is a significant decrease in the secretion of both IL-17A and interferon gamma. So this can be also blocked by the lactate transporter, because lactate is secreted, then has to be picked up. There is a lactate transporter that works very similar to the glucose transporter. Unfortunately, there are not commercially available antibodies. So we got an antibody from a group in London, and is working by and large. So the qPCR data are not always telling the truth about the expression of a transporter. So here we have the 2-NBDG, we have the qPCR data. In the case of the lactate transporter, I'm not very sure that the results are showing the truth. So that's why I'm not showing, but it is possible to use an inhibitor of lactate transporter whose name is SLCA/15-3. And one of these chemical inhibitors actually is inducing the same effect. But I don't know how much expression we have in the old versus young condition. Of course, this effect and also this effect is mediated by NF-κB. So NF-κB is the prototype of inflammatory cytokine. So here we prepared cytoplasmic and nuclear extracts from CD4 coming from these conditions. So here put obese, but is young and old. And is my deformation to prefer the obesity study than the aging studies. And, you know, these CD4 are already activated after we take them from the co-culture. And in fact, there should not be any NF-κB in the nucleus, because NF-κB in the nucleus means activation of NF-κB. But it's already there without any other stimulus other than 24 hours in the presence of the old B cells. And when we use the inhibitor, you see that there is an inhibition of the translocation, which is quantified here. Okay? So B cells are regulating the generation of pro-inflammatory T cells through metabolic pathways. And one of the crucial molecules is for sure lactate, but maybe it's not the only one. Now, lactate has these effects on T cells. What happens to the B cells if we stimulate them with lactate? Again, we used B cells from, in this case, young individuals, because we wanted to show that if lactate can be inhibited in the cells from elderly individuals, because there is a lot of secretion, probably there is not enough secretion in cells from young individuals. And if we put them in the presence of lactate, we can mimic aging or, you know, obesity or these kind of conditions. So what we did here, we took B cells from young individuals and we stimulate them in the presence or absence of lactate. And then we did the seahorse, but this is a glycolytic test. This is a little bit different test. It gives you the profile, only ECAR data, and it gives you the glycolytic profile of the cells. And this is obtained by adding in the microports glucose first, that is activating the glycolytic phenotype of the measure, of the cells in measure. Then oligomycin, again, oligomycin is blocking ATP synthase. And then 2DG is an inhibitor of the usage of glucose in the cells. So the glycolysis goes back almost to normality. You see, the B cells from young people without lactate are not so glycolytic. If you put them in the presence of lactate, whoop, the glycolytic phenotype goes up. So they become glycolytic. And at the same time, if we measure by fluidyme the expression of glucose transporter, the enzyme of the anaerobic glycolysis, as well as another enzyme of the mitochondrial function, all these are upregulated. Again, more these two as compared to this. But are we really making young cells looking like old? Well, the answer is yes, because these are the B cells from elderly individuals, untreated with lactate. They are absolutely overlapping with the young B cells in the presence of lactate. So this is one of the molecules probably that we have to inhibit in vivo. There is no small molecule that can be given in vivo at this point. Can only be delivered, either delivered specifically with aptamers, with nanotechnology, or something like that in situ, or cannot be given orally because apparently these compounds are still with a lot of toxicity, and so they have to go through preclinical through the phases of preclinical trials in order to be approved. So, and also here comparing by fluidyme the expression by qPCR of these markers. Again, you see that the B from young individuals in vitro with lactate are extremely overlapping with the ones from elderly individuals. And in addition to these markers that are metabolic markers, all the SASP, all the senescence-associated secretory phenotype markers are upregulated in the presence of lactate and similar to what happens in B cells from elderly individuals. So actually with lactate, we induce, sorry, Valkyria, we induce a senescent phenotype in the B cells. We haven't done the same analysis with the CD4, and the reason is that, as I showed you before, the B cells are very high in expression of SASP markers before any kind of stimulation. The CD4 are not the same. You have to stimulate them. Their difference, metabolically speaking, is not as big as the B cells. But if you try to do something like that on the CD4 or CD8 without stimulation, the data are very overlapping. So there is not a big effect. Of course, in these conditions, there is secretion of autoimmune antibodies. Here we are measuring anti-double-strand DNA, anti-nuclear antibody, more in the presence of lactate. So these cells are actually pathogenic, as well as it happens in the elderly. Now, senolytics, that's the final part of my talk. You know, senolytics, these are the results published by the group of Kirkland. Everything comes from Mayo Clinic for the results on senolytics. So here shown in mice that you can induce senescence just injecting senescent cells. These are induced by X-rays, by radiation. And then you can treat these mice in vivo for 5 days with a mix of senolytics. This is dasatinib and quercetin. And then you measure the physical function of these mice that, of course, in the presence of the senolytics look younger. So they run faster, the treadmill gives a positive result, they have a grip strength higher, and all these things. They don't sleep too much, they run, etc., etc. So this is in mice. In humans, these are the results that I was referring before. This is still going on. This is a clinical trial, still a clinical trial on type 2 diabetic patients with obesity. And these are biopsies from the tite, from the obese tite, biopsies of the subcutaneous adipose tissue. And these people have taken the mix, the senolytic D plus Q mix for 14 days. No, this is just the staining. It does a biopsy. D is dasatinib, right? Dasatinib and quercetin. This is the mix of senolytics. He has a lot. He has fisetin. He has mixed them together. Yeah, no, dasatinib. Apparently, this is the best for getting rid of the senescent cells. So these people are giving a biopsy of the subcutaneous adipose tissue from the thigh before and after taking these senolytics for 14 days. And this is the results. He's measuring the P16, which is a marker of senescence and cell cycle inhibition, and the typical senescent marker, which is the senescence-associated beta-galactosidase. Unfortunately, a month later, from the beginning of the therapy, the majority of these people have again the senescent cells in the local biopsy. So the point is that the people can get these senolytics intermittently. But again, if the adipose tissue is giving an imprinting to the cells that are resident, if you remove them and then you have them going back and then you remove them and then you have to going back, you know, the bottom line is that these people will never improve fully, in part, which is, of course, something. And in fact, with these treatments, he was able, I think this is a different, no, it's the same one. Yeah, it's the same clinical trial in which he was able in 9 people, again, these are type 2 diabetic with obesity. He was able to reduce with the senolytics a bunch, you know, that's the fold change after 14 days in this treatment. He was able to reduce significantly the majority of markers of inflammation associated with obesity. So a positive result, although not definitive, not conclusive. Okay, so what we did was to measure in our cells, in our B cells, the beta-gal positive cells. It took us some time because this is a kit that is commercially available. Never trust what the kit tells you to do. You have to adjust it because we work almost 3 months on have it working, because there is a substrate of this beta-gal that actually has to be treated in a certain way that is not written on the protocol. But anyway, we were able to do flow cytometry and evaluate senescence-associated beta-gal positive cells that are higher in the elderly than in the young. And this occurs by either MFI or frequency of positive cells. And we wanted to be sure that we were dealing with some senescent cells. We were not able to sort the cells based on their phenotype, the beta-gal, but we sorted the total B cells and we measure some markers of the SASP. And you see that in the presence of the mix, dasatinib and quercetin, all the markers of the SASP were significantly reduced. It's just a limited number of subjects. This occurs very quickly because it's only an overnight, but in vitro everything is different. So we don't have, of course, any possibility to do in vivo studies. First of all, because everything is regulated by the Mayo Clinic and Kirkland. You have to be their collaborators. We are not their collaborators. And in addition, the IRB doesn't allow to do any in vivo treatment, unless it is metformin, which is the only thing that I can do. I can recruit people that have been treated with metformin following the referral of their medical doctor.
Side effects on these two periods treatment? There were any side effects?
Well, not because these are already in clinical trial. They are not. I don't know the original data. This is a mix. You can buy now the drugs, the senolytics, through Sigma, through these compounds, through these companies, and you can put them in vitro. You can test them separately. I didn't have the interest in doing the two separately because this is apparently is one of the best to prevent the effect of inflammation due to the presence of continuous presence of senescent cells. Fisetin is much better to prevent and prolong the appearance of cancer, but it's a different compound. And for many of these compounds, the structure is not known, of course. This is protected by patent. I cannot give them. I cannot give them to the people. I cannot have approval from the IRB. Oh, well, of course, if James Kirkland, is willing to do an experiment like that, he will do it. Oh, yeah. Yeah, so it's a lot of money behind senolytics. Because you can treat them. Here in Brazil, you can treat patients with quercetin. Yeah? You can use some. Well, in the US, again, they are sold. When I'm teaching my students, I show the picture taken at Walgreens at the pharmacy with all the senolytics, one up close to the other. And they're sold in the vitamin shop, in the pharmacy. Well, I don't suggest people to take it as it is, even if they give it to you. When I got COVID, they gave me some, I think it was quercetin, but that's super very low dose, only 5 pills for the 5 days, and it was a very mild COVID. But if not, you are supposed not to use them without a very strict control from a doctor. And we don't have any possibility in any place of the US, even if you collaborate with him, the study have to be done in Mayo Clinic. Now, of course, I can ask you, friend of mine, to take dasatinib and quercetin, then get the influenza vaccine, and then give me the blood. But, I mean, I would not do anything like that. It should be of beneficial effect, and they don't have any study on effects of vaccine. The only study they did in COVID, and Christina Kamel was the first author. Christina was first in Yale, and then she joined his group, was to evaluate in mice the reduced mortality against the virus. So they used the coronavirus to infect mice. And then these mice were given fisetin for a long period of time. And they saw that the treatment was able to, first of all, reduce mortality in mice. And then also evaluating the effects, she was seeing something like that, this significant decrease in mice treated with fisetin in all the SASP markers like P16, P21, P53, all the cytokines, et cetera, but was a study done in mice. They haven't done anything, at least until a month ago, because I asked him, nothing in vivo on vaccine responses. And with this, I think I'm done. So this is I always like to have models because the reviewers of grant like sometimes not to read the grant and see the model. So when we have healthy aging, we have, of course, dysfunctional immune cells, and of course, low immune cell function, which means increased propensity to diseases, increased probability to get infected, reduced responses to vaccination. And this is due to the fact that there is, first of all, a lot of glucose around. Glucose is an immunosuppressive compound. So if you take immune cells and you put them in a culture medium together with glucose, even at low molar concentration, you get every kind of inflammatory marker that is product that is secreted. So, but glucose transport is upregulated because there is a lot of glucose around that is picked up by the cells. And through this pathway, pyruvate, you have increase in lactate, increase in mitochondrial dysfunction. And you have, I didn't talk about the fatty acid oxidation because it's another pathway that can be evaluated. You know, there are a lot of fatty acids that can be just picked up and oxidated through this pathway, through the enzymes like acyl-CoA, carboxylase, and all these kind of enzymes that can convert these ones in energy. And of course, after this metabolic activation that is associated with aging and senescence, the immune cells express a lot of markers associated with inflammation, immune activation markers, ASP, and of course, all the transcription factors for good antibody responses and good immunity are downregulated. And the idea is to treat them in vivo mice at this point, or Kirkland, with senolytics and or metabolic inhibitors. And metabolic inhibitors is not only metformin. There are several others that can be tested in order to improve and revert this situation from a very red situation to a blue, in which, of course, these pathways are downregulated, less activity of the lactate, and of course, less inflammation and addition and improved immune cell function. And this is my last slide with a summary. Again, the aging changes the metabolic serum phenotype of an individual. The strong data have been done only in mice so far, at least to my knowledge. And aging increases the metabolic markers associated with inflammation, and these, of course, are negatively associated with antibody responses to SARS infection that they tested and to influenza vaccine. Aging also changes the metabolic phenotype of immune cells and makes B cells hypermetabolic and also capable of inducing pro-inflammatory T cells. One of the molecules that is used in this pathway is lactate, that is inducing immunosenescence in CD4 as well as in B cells, making them similar to those from elderly individuals. And then we can have some hope that senolytics can be used soon also in vivo, not only in vitro, in order to block the SASP and improve immune function. And that's it for today. Thank you so much again for your attention.
Thank you again, Daniela. Very nice presentation and very exciting results. So
yeah, I'm always non-metabolic excited. So probably because I'm getting older, so I'm hyper every day.
So any questions? I think we have online questions.
Thank you, Daniela. Nice data. You show that in B cells, you have increased glycolytic ECAR, and then also increased oxidative phosphorylation, isn't it? Yes. How is the control? Because in normal cells, we have a switch to the...
Yes, I was expecting the same. I was expecting absolutely the same. I was happy seeing that the glycolytic pathways were upregulated more than the oxidative phosphorylation. But actually, this is healthy aging. You need to have a disease. You need to have a condition. For example, HIV. I have some donors or results from HIV patients. Well, those are only like you don't see absolutely the activation, the minimum activation of the. And I think the same, for example, is when you measure the infection with the virus versus the vaccine response against a viral disease. So you need to have the big hit of the stimulus in order to have this complete separation of it. But still, in these healthy subjects, I have more glycolytic than oxidative phosphorylation. I'm happy.
Yeah, but these two pathways stimulated one of them to produce energy and other to produce lactate as a signaling molecule.
You need, I think, energy anyway to support the development. Some energy is needed because they have to proliferate, they have to circulate. So some energy is needed. Lactate doesn't make apoptosis. So lactate is just a way for the B cells to induce other inflammatory, to activate inflammatory pathways in the B cells as well as other cells. But I think that the two of them have to be present at the same time. And even if we measure the fatty acid oxidation, we have another way, an additional way to generate energy. Energy is needed from the naive to the terminally achieved cell in order to support the basic metabolism. Then what is prevailing actually is what is induced based on the signals that the cells receive from the environment. That they can contribute to, or they can receive, just because other cells are contributing. But I've never seen this black and white situation in the B cells from healthcare. Even the B cells from breast cancer are similar, allow glycolytic function, but still the oxidative phosphorylation is active. Again, I think you need a very big hit in order to see the black and white.
From the online, just a second. Yeah, it's from Amanda. She's nowadays on Lord's Lab in Birmingham, UK. She's watching us on YouTube. So she says, brilliant presentation. Which other circulating metabolites or other changes in older or obese individuals or mice could increase B cells glycolysis and therefore SASP if lactate is not higher in their circulation? This is the first question.
Well, that's a very good question. I said before that lactate is one of the metabolites that I have studied and I have evaluated in this context. But I don't exclude the contribution of others. Now, you are in a lab where where you don't study B cells, you study neutrophils, you study other cell types. So the situation can be different because you don't see it high in serum, and this may be related to the condition of the person. I think they're healthy. Can be, if you have seen my mouse data, mice are different, even if the majority of them show high lactate dehydrogenase, some of them don't. So it can be variability among individuals. But the changes in the metabolic pathway may be due to other pathways. Maybe lactate in these individuals, in these cells, is downregulated for reasons that I don't know. So it's an open question. Interesting. Thank you.
So she continues asking, would it be just the usual higher glucose levels and lower sensitivity to glucose and insulin with aging?
Yes, it is exactly what happens. They have higher levels of glucose transporters. They pick up more glucose and they activate inflammatory pathways. One of these, maybe through the polarization of CD4, is the release of interferon gamma, which is the cytokine for insulin resistance.
Thank you, Amanda. Thank you, Daniela.
Hi, a very nice presentation, thank you. Do you know which is the signal inducing the metabolic changes in B cells? Which is the first signal? Yeah.
Well, the nutrients, signals that coming from the diet, that come from diet can be glucose, can be fatty acids, can be the increase in some amino acids. And you see also in the slide of the double negative B cells that are activating spontaneously the AMPK kinase. There are a lot of things that are responsible for this spontaneous activation.
Environment.
Well, the old environment is an hypoxic environment, is an environment in which you have an unbalanced calcium regulation. You have a lot of oxidative stress, and oxidative stress is releasing, for example, lipids. And so I think there is not one signal. And I don't think there is one that is preceding the others. Again, is a mix of signals that are responsible for activation, that converge on a major pathway. Again, it's not the only one. It's never one or the other.
Yeah, thank you. And about your first presentation, you said that the adipocyte tissue present a memory when people lose weight.
Mm-hmm.
You said that there is some memory. Do you think that this memory is an epigenetic memory?
Oh, I don't know. This would be very nice to evaluate. I don't know. Well, the imprinting of the adipose tissue to the immune cells, maybe I think that there is a great probability that can be epigenetic. Yes, although I think there are some data on the effects of obesity on epigenetic pathways. Now, when you lose weight, how much of these are maintained and how much are lost? I think that nothing is reverted completely. So yes, maybe, definitely is. My bet, I don't know so much of epigenetics, I apologize, but I'm pretty sure that there are data in the literature about this.
Thank you.
You're welcome.
Thank you for your talk. And I was talking to you about our project in immunometabolomics. It's very simple. But besides amino acid quantification, we would like to see glucose, lactate, and maybe do some correlations in serum. It's a very simple alternative for what you show here. What do you think about this?
Well, I don't know. And this is maybe a clarification to the question from Amanda. So I am not sure that, because I haven't seen any measure of lactate in serum. So I don't know how much lactate is regulated in serum. This is only made in cell culture. And there is a very sensitive ELISA that can be measuring lactate. In serum, you can measure lactate dehydrogenase. That's very strong and solid evaluation. Lactate, I haven't done any measure, but I haven't seen anything in the literature. So probably, Amanda, this can be the first data to show lactate presence as a metabolite in serum from people exercising, old people exercising or not. So I suggest you to look as a better marker at lactate dehydrogenase. And there are kits that are affordable, they are reliable, so can be found easily. So you don't need to go into mass spec in order to measure lactate dehydrogenase.
Lactate is also secreted by erythrocytes.
Excuse me?
Lactate is also secreted by erythrocytes.
Yes.
You don't have mitochondria to liver.
Yeah, that's another level of complication. It is out of the picture here. Let's talk only of the immune cells. It's already complicated. Yeah, but I don't know. Do you have experience of measuring lactate in serum? Do you know if there are papers measuring lactate in serum? I haven't seen them. Yeah, you don't know how many inhibitors of the metabolite are in serum. That's a problem. That's the problem, I think.
You just said that on T cells is more complicated to do this sort of assay because on T cells you have to stimulate them to, yes, yeah. So how, you have any idea how this activation can interfere with the results you get? You have an idea? Because it's something else you adding.
Well, I'm talking, when I said that, Valkyria, was when I was measuring by seahorse, the OCR and ECAR in just sorted ex vivo isolated cells, okay? In this transwell, the cells are activated by B cells. Who knows which stimulus is coming from the B cells? So we say that is a metabolic activation, okay, but what is the B cell secreting? Probably they secrete lactate, probably they secrete other metabolites. I mean, when I was talking of the seahorse data, those were really without any stimulation, but the others are.
This one without stimulation, because we're going to do T cells from the dog into mitochondria and so on and so on. So if you don't stimulate the cells, can you see any difference or
well, I have a little difference, which is not as much as the one that I've seen in B cells. So I don't know in these individuals, the situation can be completely different. And if not, you give a mitogen, you give an anti-CD3, you give a PMA, even a cytokine can be, you do IL-2, can be useful to see the seahorse after the activation of the CD3, I think total CD3. But again, the healthy aging can be totally different from the samples that you want to evaluate. So you have to run them first and see if actually the two groups show a difference.
You are trying to compare young and more than 6 years old individuals and look at mitochondrial function on T cells, on phenotypes of T cells. But now that you said that we have to think more about this, if we have to stimulate, because we saw differences anyway.
Well, don't forget that I have differences in CD4, not as big as in B cells. Nothing in monocytes, for example. So a difference, you can see it, it's not so, yeah, so big.
Because we're doing some pilot experiments just to, yeah, okay, thank you. Questions?
Well, thank you, it was very good. Thank you, Valkyria. Thank you, Diane. Oh yeah, I forgot.
I took from your first presentation.
Yeah, but that's the second one.
Okay, so go there again. I'll take the second. Immediately. I took from you. No, just to show copies. Maybe you just take this from Danielle.
No, this one, because this is for the University of Miami. They have to put it in the newsletter. Any international presentation. Okay. But not like Claudia Schiffer. I mean, like, right here. With a different type.
Yeah.
Perfect.
Going to go there with the
Yeah, so we can put everybody on the
yeah, let's hope you take everybody presentation. So we will have your presentation.
Okay.
Is that on the, it's not. No. Let me see now. So, one picture of yes. Here. I'm putting a picture of this. More and more and more. All file. Thank you. Check it. I think you want to get a lot of great things. Oh, I thank you. You were very good. I'm pretty sure that means that all of that.
E aí eu só pensei nesse grupo legal na conversa. Muito obrigada. Lá que eu acho. Thank you, bye bye. Meu contato. Isso me sente meu perfil. Não, super ativo, super.
Eu é segunda-feira, da tarde. E uma vez por ano, uma vez por ano, a gente tinha um protocolo com uma avaliação geriátrica e falando, né? 2 horas da entrevista, o teste. Então é super interessante porque você vai ser comparado com você mesmo. Você não tem que comparar com ninguém.
Como o número de melhorar,
tudo que você melhorar, tudo que você